is quite good for these patients. So that's the patients. The second issue is, of course, safety. So we compare the different products. We really want to see the, the efficacy. Also, we want to see the safety issues. So it's totally surprised to us, actually, it's quite safe, not like, cancer, because we can see that, apart from leukemia, because we do the precondition, and other, not much change. And of course, the neutrophil, they will come back after a couple of weeks. And we didn't see the fever and a few other things because, and let us to understand, because the, the loading, B-cell loading in autoimmune disease is not that heavy, not like a cancer. And patients, the burden is very heavy. So we didn't see the dramatically increase in cytokine. Also have one or two cases have a little bit high levels IL-6, but mostly, and most cases, it didn't have much change. So let us feel very satisfied for this treatment for the patients. So that's another lesson we learned, because different disease, cancer, autoimmune disease is quite different in many ways. So, and finally, I really like to acknowledge my students and my staff, and in addition to those collaborators I already mentioned. So my staff is a worker, they have to do the clinical work plus research. We trained our team just like a physician scientist, and we also have lots of students help us for some basic research. They are in this room, many from the Tsinghua University. Thank you very much for your attention. Very quick talk.
Well, maybe I'll start. It seems interesting to me that with the autoimmune conditions, the durability of the response and the survival of the cells would be more important than for cancer. So can you tell us, do you have any evidence that the cells are still surviving at these 12-week time points?
Well, yeah, the cell survival, we see that by 6 months, by 3 months, they survive very well. And so we are checking, but not published yet. So we can see by 9 months, some cells still there. But the different products are different because, like, the STAR-T, they really depends on the antigen over there. So it's slightly different. So we compare, we want to know what's the difference between products design. Some is designed based on the PCR, some is just knocking straightforward and different. So slightly different, but we can see the durability is quite long, actually. It's not compared with the autologous CAR-T, not much differences. That's another thing that surprised to us, actually.
Thank you, that's very interesting. Any other questions?
Thank you very much for this great talk, Fabian Müller from Erlangen. And I'll speak the next talk for the autologous CARs. So your data is really beautiful and striking. I have many questions. I guess the most pressing ones are, so when you look at your allo-CAR, it appears that your allo-CAR seems to persist, actually. Like the numbers are not going down to 0 in the autoimmune patients, at least for the 3 you published recently in Cell. Could you comment on that? Do you see, like, later on that they go away? And in regards to...
Say that again. Because I echo from this side. So
oh, yes, of course. Do your CARs, the allo CARs, do they persist in the patients? Or do they go away?
Yeah, at least we can see in 3 months it's already persist over there. But eventually they were gone, of course. So we checked, because some patients already reached quite long now, more than a year, we didn't see.
So they're going out then.
Yes, they go out. Eventually they were up. So that's another question to even the reviewer and a few other people ask us. Eventually they will go. That's another advantage. So we don't really concerned about the cancer because eventually they will go.
Yes, and a second question, if I may. So you, in your myositis patients and all the systemic sclerosis patients, not all of the autoantibodies disappeared. Some of the patients, the autoantibody persists. How do you think the disease is going away, even though the autoantibody is gone?
Well, but we can see for the, the myositis, the antibody is gone. So for others, I show that for the scleroderma, some is keep in certain level, but they still, compare with, before the treatment, they drop a little bit. But some is dramatically. So that's the things back to the clinic, back to the pathogenesis. How important for all the antibodies? That's another issue. So, but at least we can see they dropped. They all dropped. But we try to recruit the patients we really need based on the autoantibody. If they don't have an autoantibody, we would think about they may not relate to B cells. But you are right. We show the data. We show the 06 patients. We see some patients not drop significantly and dramatically, but they do drop a little.
Yeah, we see the same thing. So that's why I'm curious how you feel about it. Thank you.
Thank you. I think that's all the time we have for questions. So let's thank Dr. Xu again.
Thank you.
And next, of course, we have Professor Fabian Müller. He's a professor at the University Hospital of Erlangen, Germany. And the title of his talk will be CAR T Cells in Rheumatologic Disease, Lessons Learned.
All right, good morning, everybody. First of all, I want to thank the organizers for having me here. It's really a great pleasure, and it's a great honor to be allowed to present our data on the CD19 CARs in rheumatologic diseases. My name is Fabian Müller, and I'm working together with the two gentlemen at the bottom, Professor Mackensen, who is a hematological doctor just like me, and Professor Schett and his team on the room side. We together have developed this couple of different patients that I'm going to introduce to you. I want to actually start a little bit further in the very beginning of the basic biology so that you can bear with me. What is really important to understand is that a T cell normally has just a T cell receptor on the surface, and we have been hearing a little bit about it. But I want to go into depth so that you understand the biology and the beauty about what we have been doing. So a T cell receptor, you see it at the bottom, is consisting of a couple of components. And a T cell receptor typically needs on the other side, the other cell, here shown at the top, an MHC receptor. And only if the MHC is matching and this little red dot, which is a peptide, the T cell can engage the target cell and then either start an immune reaction, and if it's a CD8 killer cell, start killing the cell. We use that in cancer therapy, and we can introduce pretty much artificial T-cell receptors that then target specific mutations important for cancer cells, but still this is dependent on the MHC. In CAR T-cell therapy, the receptor looks very, very different, and the CAR, the chimeric antigen receptor, is really artificial. What you see is here that the T-cell receptor at the bottom, the SATA chain, is pretty much the same than the signaling of the TCR. You have a co-stimulation, and then at the top you see an antibody fragment, and this antibody fragment is actually directed against an antigen surface antigen, and by that we can target whatever cell we want to target. And once it's actually engaging the targets, shown to the right, you see that the T cell then, because of this CAR receptor that is firing, starts killing the target cell. And these cells have been a tremendous success in cancer and have revolutionized what we are doing at our hem-onc departments nowadays. I want to show you an example patient just so you understand what this therapy actually has, what the power is of that. This is a patient at the time who was going through all the different therapies that were possible. He's in the mid-40s, or was in the mid-40s at that time. You see in this PET-CT scan that glucose that is marked, that is labeled with an imaging agent. Everything that is black is cancer except for the brain and the gallbladder, the bladder at the bottom. So you see a high load of cancer in this patient, and he would have been dead as of now without CAR T cells. He then received a CAR product. It's the same product that we use later on. And I used that slide because I want to give you the impression what is actually happening. You infuse the CARs at the very left end, and then over time you see that the CAR is first expanding. So it's really that the CAR is a living drug. It's different from any other drug that we use in the clinic. So the cells are then expanding, attacking the target cells, in that case CD19 positive cells. And you see that pretty much any T cell in the blood of this patient at like 2 weeks after CAR infusion are CAR T cells. It then leads to a contraction phase that is typical for any T-cell directed immune reaction, and we see that also on the other end we have a persistence up to more than 3.5 years now. At day 90, when there was like roughly 100 cells per microliter CAR, still the tumor was entirely out, and the only thing you see here is like the heart and the urinary tract system again. So we see these tremendous responses in approximately half of the patients where we can control the disease then for the rest of their lives without cancer. Next to the success in cancer, we started then thinking about other diseases. You see here just a few of the examples that are happening nowadays. On the left, you see fibrosis, so we can prevent scarring. On the right, senescence, we can actually turn around aging of certain cells. And we can talk about autoimmunity, where you see we can target B cells in the center. We at our center, we have decided to use the same approach that we just have heard, the CD19 targeting of B cells in the system. I brought you this slide to kind of illustrate a little bit on the mechanism behind it that we today think what is happening. You see in the center a B cell, and the B cell is always the B cell receptor on the surface, that's the antibody, and it detects the autoantigen, and because of that binding, the B cell starts to be activated, produces antibodies, produces cytokines, that then leads to an immune activation for these B cells. On the other hand, you see to the right the T cell, and the B cell is also the antigen-presenting cell. So you see that this antigen that it binds through the BCR is going to be processed and put on the MHC, and by that it can be cross-presented to T cells, and that ultimately leads to priming of the autoreactive T cells as well. So the B cell is really central. By taking the B cells out of the system, you can appreciate that we likely have a couple of different ways of how, the immune system could change after taking those B cells out. If you think of it again with the similar B cells that are activated through 3 different pathways shown to the left and to the bottom, you have to think of it in activated B cells and then the plasmablast and plasma cells, all of which are the B cells that are capable of producing antibody. If you want to target these, you can use a couple of different surface antigens shown here. Traditionally, we have been using CD20. CD20 is only targeting the activated B cells, and we at our center, similar to what we have just heard, have then decided to use CD19, which is much broader, and BCMA only targets the 2 rightmost cells. Lupus is probably the prototype of these diseases because we have suspected that the B cells and the autoantibodies are driving the disease for pretty much 30 years now. There's lots of data on it. But the field has never been clear if it's really the B cells, and that is because we see this very broad activation of the immune system with T cells, macrophages, antibodies that are there, the cytokines that are brought. What we have known so far is that there is an extrinsic factor, a precondition. The patient gets this disease. It's activated by some reaction, pregnancy or infection or something else that leads then to an organ damage, but it's usually associated with a B cell receptor, so with an antibody. There's a couple of rheumatologic diseases we just heard about, also those 3 that I'm going to be talking about, but there's many others. There's neurologic diseases, hematologic diseases, and I'm just coming from ASH where we have heard a couple of other diseases on top of that treated with CD19 CARs. So the field is really exploding right now. Our current hypothesis is the following, based on what we have done and many others have done now also. We have the autoimmune B cell, we give the CARs, that then leads to this abrogation of the B cells very fast. The CAR is then out of the system at one point and the B cells come back, and that's then a reset of the B cell. And pretty much the basic idea is, or the most important point is, that we believe the CARs are just much deeper going inside the tissue because they actively invade and not just passively diffuse, and by that lead to a much deeper reset of the B cell compartment. To achieve that, we have used a fully automated closed system. It's the Miltenyi Prodigy, and we have been using the FMC63-based 4-1BB co-stimulated CAR shown in the center. It's a closed, fresh in, fresh out system. It's really nice because the production is very durable and very, very sturdy. So we produce pretty much all our products. It's produced within 13 days, fresh in, fresh out, as I said. Lymphodepletion, similar to what we just have heard with fludarabine and cyclophosphamide, typically 2 days of a pause and then infusion of 1 million CAR Ts per kilogram body weight. We have been treating the same 3 diseases you have heard. So we have been treating the lupus, systemic sclerosis, and myositis. I think we were the first for any of these diseases to actually treat the patient for their autoimmune disease. There were a couple of other reports now popping up where they had lymphoma or leukemia and the autoimmune disease also disappeared. I want to start to introduce our very, very first case. That was a young lady, 19 years at the time, and she had very many different symptoms. She had a walking distance of 10 meters. She couldn't sleep laying down, so severely sick. What you see here are the different different symptoms she had. And then she had very many, all the different available therapeutic options. And you see that the autoantibody never disappeared, the proteinuria never disappeared, and because of the antibody that is then using up complement, you see at the bottom left the C3, so that's complement that is used up continuously, never normalized throughout the entire treatment, and the B cells were pretty much never gone. So despite all the different treatments, we could not get rid of her lupus, and at the time we discussed with her whether she wants the CARs. She had pretty much no treatment options and a life expectancy of less than 3 months, definitely less than 3 months, probably even less. You see here, she got the CARs infused, and just as the patient I've shown you earlier, she expanded. At one point, there was roughly 30% of the CARs, of the T cells in her blood were CAR T cells. They contracted, similar to the patient that I've just shown you. And what really was striking to us and to many others is that the B cells were gone, so for the very first time in the entire career of her disease, the B cells were really out of the system by day 7. You see here also that the autoantibody was gone within almost a month. The C4, C3 normalized, the proteinuria went down to 0. So this was a tremendous success for this patient. You see here this SLEDAI, the SLE disease activity index that we just heard of. It's a composite score of these different things shown to the right. And you see that pretty much everything that this patient has was entirely reversible, which was very new to the entire field. We have published in February this year with cutoff of October last year, 15 patients. As I just said, I'm coming from ASH where I presented the 35 patients. And I want to share with you the clinical data on our now 35 patients. What you see is that those 35 patients are a couple of different diseases as shown here with the typical age distribution and gender distribution. We have a mean duration of years before the disease, prior treatments, and the follow-up since the treatment. I'm going to talk to you about safety and about efficacy, just as you've heard before. And these are 2 different cohorts with 35 or 28 patients respectively. What we have found in all of those patients was that the CAR expansion is very typical. What is intriguing is that we have a minimum CAR expansion of 3 per microliter and a maximum of almost 3,000, so that's a 1,000-fold difference in these patients. The median is about 140 per microliter, so they expand typically and they contract, and they are all out of the system within 3 months, similar to what we have just heard. We have had no manufacturing failures for any of the patients treated so far. I want to start with safety. You see that the safety is really good. Typical safety issues are the cytokine storms produced by the CARs and then the other immune cells. And in heme, we really see deadly events, often grade 3, grade 4. And in those patients that we have treated here, we have pretty much predominantly grade 1, some grade 2, and no higher toxicity. Neurological toxicity is in 1 patient of these 35 and not more. We see bone marrow toxicity. That's a typical CAR T cell toxicity. It's interesting. We call that today ICAT, so immune effector cell-associated hematotoxicity, which is not only the depth but also the length of the toxicity. You see that we typically have early toxicity, that is lymphodepletion plus CARs. It's not unexpected. And we have some late toxicity, which is predominantly in SLE, and it's always just the neutrophils that are gone. They're always responsive to GCSF, and most of the time you need 1 or 2 shots of GCSF, and that's it. So it's granulocyte stimulating factor pushing neutrophils. We did 2 bone marrow biopsies in 2 of these patients, and they didn't show any signs of blast dysplasia or aplasia. We have some infections, here shown the viral infections, needing hospitalization, but the different viruses that caused the infections, you see here, there was no ICU treatment and no severe, more like grade 4 reaction. As discussed earlier, the B cells are completely abrogated, so in all of these patients, there is no B cell detectable in the peripheral blood anymore, and in all of these patients, they come back starting from day 45. Despite the B cells coming back, the patients remain in treatment-free remission. That is really tremendous. We then did look a little bit deeper into the depth of the depletion. And what you see here is the lymph node biopsies. We did that pre-CAR at the top, post-CAR, and post-rituximab at the bottom. And you see that before CAR T-cell therapy, all of these patients had a vast number of CD19, CD20 positive B cells in the lymph nodes that were entirely gone after CAR T-cell. And you see also that approximately at the same time, so 3 months after treatment, rituximab does not deplete the B cells from the lymph nodes, suggesting that the depletion within the tissue is indeed much deeper. What we also did was we then looked for follicles within those lymph nodes. So these are the germinal centers where the antigens are processed. You see that in both pre-CAR and post-rituximab, you see these typical lymph follicles, but the lymph follicles are entirely gone in those CAR T-cell treated patients, meaning that you can't see the T cells shown by Ki67 or the follicular dendritic cells shown by CD23 in those lymph nodes any longer, suggesting that the depletion of the immune system is really much deeper than just the B cells. There is more going on than just that. We have looked together with Professor Thomas Winkler, who's a B-cell immunologist at our site at the B cells, and you see that before the treatment, we have mature B cells, and they all are set to naive cells by the CD19 CARs. I guess that's not me. So there is a deep reset of B cells following these CD19 CARs. We have then looked at the distribution of these BCRs, looked whether they are diverse or oligo or monoclonal, and you can do that, for example, by V gene usage. Some of you definitely remember how antibodies are built within B cells. So we all have 120 different V genes that are used differently in the different B cells. You can just see by the distribution of those V genes whether you have monoclonal populations. If you look at our healthy individuals or SLE, myositis or systemic sclerosis, you see that the V gene usage in percentage and frequency is very similar to healthy individuals, suggesting that it's a polyclonal B cell population coming back. So they are pretty much normally distributed and normal in diversity. I'm going to keep that short because we heard it. It's the same thing. The autoantibodies disappear. You see the different autoantibodies. Similar to what we have seen earlier is for example, in the lupus patient, the anti-NUC antibody is continuously there. Also, anti-Rho 60 here and ANAs persist in some patients. So many, but not all, autoantibodies disappear. We then looked at the immune responses against pathogens that are being vaccinated as kids, so measles, mumps, and others. You see that they are all roughly persisting, and what is also interesting that with the red arrows, you see there is re-exposure or re-vaccination for COVID, for tetanus and others. You see that the plasma cells react, suggesting that those cells are still fully functional in the bone marrow. So what that also means is that the disappearing autoantibodies must originate from short-lived cells. This is now efficacy in the cohort as promised. You see that we see at 6 months 100% responses, and 100% of our patients are treatment-free, so they are off all of the immune suppressants. And they remain treatment-free in all but one cases so far. I'm going to talk about the one case of myositis in a little bit. You see the different scores for systemic lupus erythematosus, MMT8 for myositis, and the Rodnan skin score for systemic sclerosis, just as an example for the many parameters we measured. You also see the skin that is improving dramatically for a couple of different patients. Top is a lupus, bottom systemic sclerosis patient. So we have now follow up a total cohort of 10, a little over 10 months, and of the 15 patients of the New England Journal of beyond 2 years. You see that one myositis patient has relapsed. We have infused a total of 18 patients in our CASTLE trial and 17 in the named patient access. In addition to that, we did one retreatment, and I want to show you the data. What you see here is that we did infuse initially the CD19 CAR, and it expands day 7, day 10 to 1,500 per microliter, so it's a decent expansion, and then it contracts. The patient lost the CAR just as expected. We then, in relapse, we did another infusion of the CD19 CAR, and you see that there is no re-expansion in that situation. What is important is that we did treat the patient with Flu/Cy again, and we did not see CARs, and the patient did not benefit from it, suggesting that Flu/Cy is not the thing that does the treatment. We really need the CAR expansion. We then got hands on a BCMA CAR T, which we treated the patient with, and you see here the BCMA CAR then expanded again up to 100, pardon me, up to 170 per microliter. The patient did respond, and she's in treatment-free remission as of today. So she's doing well again more than 6 months after that therapy. The success, as you have just heard also from our colleague, was tremendous and spurred a lot of different trials. This is in February, publication in Nature, so the number is substantially higher now. There is NMOSD that has been treated here, a very different CAR. The NMOSD is antibodies against the myelin sheath and produces a lot of different symptoms in the patients. This is the trial chart, and you see here how all of these patients that were treated with this mRNA CAR, it's not a transduced, like a stably transfused CAR, it's an mRNA transfused CAR, how all of them benefited really well. The clinic was improving substantially. However, the follow-up is really short, and the question is whether these mRNA CARs are sufficient to actually get them in durable remission. We don't know that yet. It's not published yet. A second story was myasthenia gravis, also by a co-worker of ours, where we have antibodies against a couple of different parts of the motoric end plate. He treated a case, one woman with substantial amount of crises, and since she was treated, there were no crises anymore. It's still up to today, it's true. She had a substantial increase in arm holding time, a walking distance, so the patient is back to an almost normal life now after treatment. As I said, the CARs disappear. We discussed that earlier also, and we always wondered whether the short peak is sufficient. So there's these questions, do allo CARs do the job? You just heard it, so I can keep it short. Yes, they do. And we have a large advantage of off-the-shelf products from health individuals, as you heard. So we have seen this slide. I can keep it short. We're very grateful for these data because it shows us a lot and teaches us a lot. We have seen that they deplete the B cells also very deeply, and we see that the B cells come back and the CARs do not persist, as we just have learned. Within the skin, and that's data from the same publication, we have done similar things as of now, but that was the first one to show it. The B cells disappear, and it's really impressive how the crypts within the skin are just normalizing and come back to normal crypts. So at this point, we have a short follow-up for all of these trials, but I think it really looks promising. I want to show two things to just make two strong points again for this treatment, for cellular therapy in autoimmune diseases. The first one is a kit. The first kit that has been treated worldwide with CD19 CARs, and it works just similarly well. You have to know that systemic sclerosis and lupus are actually very common among kids. So the peak is at 20 years, and half of the patients in SLE are 20 years and younger, and it starts from 10 years. So this is a kid who was 15 at the time of diagnosis, or when we treated her. She was 14 when we started. Within 8 months of onset, she had all treatments available, and the kidney worsened. You see that at the right. When the kidney got worse and worse, the pediatricians contacted us and asked whether we could do the CAR-T treatment. We did in dialysis, so she went on to dialysis. We gave the CARs 2 or 3 weeks after start of dialysis, and then she had 2 more dialyses after, and since then she is off of the dialysis. SLEDAI is to 0. The point that I want to make is that with this treatment, we can reverse dramatically symptoms. We just have to be early enough. The second patient is a neurolupus patient. So this person was working for the army, and in the fall he started having a weak knee, and after 3 months he was bedridden. So it was a very severe case, and no treatment pretty much got it around. At baseline before CAR-T, you see those white spots in the brain, in the spine, and the skin looked terrible in the beginning also. And 3 months after, you see that the neurological lesions disappeared and the skin improved substantially. So here, dark blue means a lot of muscle strength, and light, there is no strength. It was really that the left leg had no control anymore, muscle control anymore. And you see in those 2 videos what happened to the patient. So the doctor is asking him to move the right leg first, and you see that the leg is moving just a little bit. And the left leg, you see in a second, there is not much happening. She then moves on. Now comes the left leg. You see there is no movement in the left leg. She then tests the reflexes. What is important, if you have a spastic lesion, you pretty much have an overstimulation of the muscles, and especially we call that the Babinski sign. If you she's tickling the patient in a second, and you see how he's spreading the toes. It's a very typical thing to see when there is a substantial lesion in the spine like that. And 3 months after, you see to the right, I can't stop the other one, unfortunately, but you see that he's walking with some assistance at 3 months, and he gradually improves over time now. So it's really tremendous how the CARs, despite the fact that he is having brain lesions and spinal lesions where people would be scared to treat with CARs, we were also, but we did a couple of precautions. He had 0 side effects and is benefiting dramatically and is gradually moving hopefully back to a normal life. This is patient 1. In January this year, with 1,000 days after CAR-T, she is back to running 5 times a week. A normal life, she's studying and recovered all her organ functions. And with that, I want to summarize that we have 100% production success with the CD19 CARs with our system. Sometimes with tapering, the immune suppression is typically well tolerated. Sometimes we see flares before CAR-T that then make troubles after. The CAR-T expand in typical fashion, but do not persist. We have CD19 CAR-T cell therapy that is very, very well tolerated, much better than in heme diseases. B cells and disease-defining autoantibodies typically disappear. Naive B cells remain dominant phenotype for 12 months in lupus, and we have had this one relapse that was retreated with BCMA CARs successfully. There's several questions that remain. What is the rate of long-term efficacy? Is this a cure for some? How much of the organ damage is reversible? I think I've shown you some of the convincing data. Are there markers that predict relapse, which is something we are learning in the near future potentially? Is there any late toxicity, which we generally don't know about CARs because we only do that for 12, 13 years now? Are there other diseases that can be treated? The answer is yes. There's many others happening currently. BCMA versus CD19, BCMA and CD19 double CAR. Do we need persistence? And what is the role of lymphodepletions are just some of the questions we have. Many people I have to thank, especially those at the top right. Melly Hagen is my partner in crime on the clinic side, our 2 bosses. Many people, as you see here, and most of all the patients. I want to thank you for your attention and look forward to the questions. I stole the microphone, so.
First of all, thank you so much for you including our study in your talk.
Of course.
Thank you. And I just wonder, do you have got a chance to check the B cells in bone marrow after the transfusion? Related to this, I'm just curious, in what circumstances you can get lymph nodes?
I think that's a very, very good question also, because what we are currently trying to understand is the disappearance of the B cells. When do the T cells come back? When do the CARs go away? All of that is very closely intermingled. When we look in the bone marrow in CD19 CAR T cell treated patients at various different spots, as long as there is CARs, there is pretty much no CD19 positive cell in the bone marrow in our experience. That's what we have seen so far. For the autoimmune patients, typically the CARs are already in contraction, but we still do not see yet CD19 positive cells. And I mean, it's only 2 cases that we have done, right? And you would have to look in more detail, in more timely manner to understand the dynamics in detail. But as of now, it's no CD19 positive cells in the marrow.
In what circumstances you can get the lymph nodes? Because it's not easy to get the lymph nodes for these patients.
Yeah.
So how did you get the lymph nodes?
So the
where is the lymph nodes?
Yeah, it's like in the inguinal tracts, okay. Right? So where you have, as a normal being, like all of us have lymph nodes of like 1.5 centimeters here. So you can do it with ultrasound guided biopsies. You need a very trained and a skilled person to do that, but it works.
A great talk. So I have a very naive question. So for this autoimmune disease CAR-T, do they produce the same technique as CAR-T for B-cell lymphoma?
That's not a naive question. It's probably one of the more crucial questions. Thank you for that. So we produce with exactly the same, like the same machinery, the same everything, the CD19 cars. For some of the pharmaceutical companies, they are trying different things. Sometimes it seems difficult if you use frozen material, which many people do. If you use frozen material, it sometimes can be tricky to get the cars actually to work.
Okay, so do you test the percentage of CAR positive cells before you transplant them? Do you see any correlation of this, like the percentage? Because in the CAR T for B-cell lymphoma, the correlation is not very clear. So for your treatment, do you see any kind of this correlation? Like you have higher percentage of CAR T positive cells, you have quick response and relapse.
So there is no correlation that we could establish so far. I mean, the patient numbers are still fairly low, and the different diseases behave very differently. For example, a systemic sclerosis patient, they experience no side effects whatsoever. So you can do with them whatever you want, honestly. It's the easiest of all of them. Whereas with the lupus patients, sometimes it's a little more dynamic, the reactions that they show. So we need more numbers to say that. I can tell you that the percentage of transduction is very similar in our hands. The phenotype of those cells, we were just writing up that paper, is actually different. We see a different distribution among the T cells in the product regarding naive central memory effector cells and effector memory cells. So there seems to be something else which might contribute to the beneficial side effect profile that we see.
Yeah. So quick last question. Okay. So I see like you have shown one piece of slides. You show like the vaccine antibody remains high. Did I remember correctly?
That is exactly correct, yes.
Yeah, so I'm wondering why the vaccine antibody remains high, but the antibody against this AID antibodies seems like eliminated. So why is there selection?
That is really crucial to understand why I believe the CD19 target is a good target. And with you asking that question, I wasn't being clear enough with it, so I apologize for that. So if you remember one of the earlier slides, I showed that there is BCMA and there is CD19 as potential targets. BCMA targets plasma cells. The plasma cells are those residing in the bone that are producing our antibodies, right? So the long-lived plasma cells. They are CD19 low or negative. There is an ongoing argument about it. So those we just don't tackle with the CD19 CARs. If you give a strong BCMA CAR, you get rid of those antibodies. So if you use BCMA, you might actually deplete also those B cells, which then means that you could get rid of the autoantibodies that come from these long-lived plasma cells. For lupus, we don't need that. CD19 is totally fine. For the others, it's not clear yet, and we need longer follow-up, if you ask me. Thank you for the question.
the McLean Hospital at the Harvard Medical School. And today he'll be talking with us about autologous cell therapy for Parkinson's disease, from bench to bedside and from bedside to bench. Dr. Kim.
Good morning, everyone. First of all, I also like to thank Sheila and other organizing committee members for inviting me to this really fascinating symposium. And unlike the previous three speakers, I'm a basic scientist. I'm not an MD. So maybe we can change the perspective a little bit. That's why I put my the talk title, autologous cell therapy for Parkinson's disease, from bench to bedside and from the bedside to bench. As you may all know, the Parkinson's disease was identified in 1817 by Dr. James Parkinson. This is the original article which he described this neurological disease as a shaking palsy. And that is like more than 200 years ago. And a lot of studies have been done. And this is typical posture. And despite the enormous studies, we still don't quite understand the underlying pathological mechanisms of the Parkinson's disease. But one thing very clear is that a hallmark of Parkinson's disease is age-dependent and selective degeneration of A9-like dopamine, midbrain dopamine neurons in the substantia nigra, which causes motor deficits in Parkinson's disease. Currently, there are about 10 million Parkinson's patients over the world, and with the aging population, the social burden is becoming bigger and bigger. So because of this very specific pathological hallmark, single type of neurons is causing the major motor deficits in PD. Cell replacement therapy is emerging as a very promising, and in fact, Parkinson's disease is considered to be an ideal target for cell replacement therapy. And since 1980, numerous scientists have tried the transplantation of different types of dopamine neurons in diverse animal models and in patients. Among these cell types, they found that transplantation of fetal midbrain dopamine neurons from aborted fetuses provided the most promising outcomes. So in early 1990s, pioneering scientists like Dr. Ulla Lindvall or Anders Björklund in Sweden pioneered this type of study using fetal cell transplantation and reported really amazing results in those patients. This is open label using very small number of patients. And those results were published in the top-tier journals as well as in hundreds of other scientific journals. And some cases, even after 1 or 2 decades, after one-time transplantation, the patient's improvement sustained. So encouraged by these initial open-label promising results in USA, 2 groups performed large-scale sham-controlled double-blind study, and they published in 2001 and 2003. And unfortunately, their conclusion was that clinical improvement was variable and insignificant, and they did not recommend any further fetal cell transplantation. Furthermore, in those 2 studies, about 25% of the patients developed very terrible side effect, which is called graft-induced dyskinesias, which eventually terminated this approach. So in summary, fetal cell transplantation studies demonstrated the proof of principle of cell replacement therapy for Parkinson's disease. However, fetal cell transplantation has a significant ethical, practical, and medical limitations to become general therapy. In fact, some neurologists believe that cell replacement therapy in Parkinson's disease is a failed approach and should not be pursued. Actually, this striking comment was what I got when I applied NIH grant 3 or 4 years ago. This was one of the 3 reviewers. So after the trials and errors of the fetal cell transplantation, more recently, people are focused on using the human pluripotent stem cells, such as embryonic stem cells or iPS cells. And as I briefly shared in the preview in Cell Stem Cells several years ago, there are several different types of approaches. One approach is using the human embryonic stem cell as the off-the-shelf product, and people differentiate the dopaminergic progenitor cells and use them as the off-the-shelf product to treat Parkinson's patients with immunosuppression. And this is maybe one of the most popular approaches these days, being performed by several groups over the world. And another approach is starting from the donor, we can generate human iPS cells, clinical grade iPS cells, and dopaminergic progenitor cells, and do the same way. And in some groups, like Dr. Jun Takahashi in Kyoto University, they're approaching using the HLA-matched donor and then biopsy and iPS cell generation and dopaminergic differentiation and treat HLA-matched Parkinson's disease patients. This trial has already started, and we expect to have the results soon in summer. And then finally, the other approach is that starting from the patients, then biopsy and generate the clinical grade iPS cells and differentiate, and then go back to the patient brain and then do it as a personalized or autologous cell therapy, which is our focus. So about a decade ago, we decided to pursue the autologous cell therapy and identified key issues to establish a platform. 6 issues. Number 1 is obviously safety issue, especially to get FDA approval. We have to make sure that it is safe. And then second issue is the reprogramming technology. Although Professor Yamanaka did amazing invention of the reprogramming technology, this is still premature and the mechanisms are still unclear. And then number 3 is in vitro differentiation for scalable production of transplantable cell sources. And number 4, once we make the functional dopaminergic cells, we have to validate there is safety and efficacy using valid animal models. And number 5 is the neurosurgical device optimization. And lastly, we have to take care of the regulatory, budgetary, and the clinical issues. So I'll very briefly share with you our research to address these issues. The number 1 is a safety issue. When we talk about the safety of the stem cell-based cell therapy for Parkinson's disease, we have to consider 2 issues. Number 1 is there should be no remaining undifferentiated cells, because by definition, any undifferentiated iPS cells can generate tumor. So luckily, we found that undifferentiated iPS cells or human ES cells express very unique set of anti-apoptotic gene, which one of them is BRC5, which encodes survivin. And survivin is a very famous gene among cancer research, and they already developed very effective inhibitors such as quercetin or YM155. We found that when we treat the cell, differentiating cells with quercetin for short time, such as overnight with a low concentration, we can eliminate, kill undifferentiated iPS cells with greater efficiency, like more than 99.99%. In contrast, when we treat the quercetin to the differentiating cells, like precursors or neural progenitor cells, they are not at all affected. So using this fact, we have a scheme that we differentiate in the middle of the differentiation. We treat the quercetin for overnight, and then we can completely remove any remaining undifferentiated cells. And then a second issue for the safety is the genomic integrity must be confirmed by, we should confirm the normal karyotype, and there's no host chromosomal disruption and no residual DNAs, and no harmful mutations by the whole genome sequencing or whole exome sequencing analysis. Number 2 is reprogramming. And when we try to understand the mechanism of the reprogramming in greater detail, we focused our attention to the very interesting phenomena, which is called Warburg effect. And you may have heard, learned about Dr. Warburg's work in the textbook, right? So he's the amazing biochemist, and about a century ago, he found that when normal tissues become cancer cells, their metabolism is completely changed from the oxidative phosphorylation and glycolysis. And this is called Warburg effect, and amazingly, the same phenomenon was observed when normal tissues become iPS cells during the reprogramming process. So we call it reprogramming, metabolic reprogramming, and we try to understand the mechanism. And we found that NAD-dependent deacetylase, SIRT2, is dramatically decreasing during the reprogramming process by the microRNA 200C, which is induced by OCT4. So OCT4 induced microRNA 200C, and microRNA 200C decrease the SIRT2, and as a result, most of the enzymes among the glycolytic pathway is hyperacetylated, and as a result, the enzymatic activity is activated, resulting in the enhanced glycolysis. And we also found out that the same pathway, microRNA 200C, SIRT2 axis, also regulate oxidative phosphorylation by triggering the mitochondrial remodeling by two pathways, which is MEK1, ERK, and DRP1, and then AKT1, DRP1 pathways. And as a result, the same pathway controls this metabolic reprogramming. In addition, we found additional microRNAs that are also involved in this the metabolic reprogramming process, and we also identified combination of the optimal factors using Yamanaka 4 factors and 2 of the microRNA clusters, and we established this second generation reprogramming method. And number 3 is the differentiation, and most people use the monolayer culture, and we found that when we culture and differentiate iPS cells using this monolayer culture, they easily detached, and then many apoptotic cells come out. So we invented a novel method, which we call the spotting-based method, which is basically growing and differentiating the cells in a smaller portion of the plate. So as a result, we found that this spotting-based method can generate much healthier and functional cells. And then after we differentiate the cells using our spotting-based method, we tested their functionality and safety and immunogenicity. So when we tested the differentiated cells using our method, they did not produce any tumors, and also they did not produce any outgrowing cell grafts. And then also using the very popular 6-hydroxydopamine lesioned rat model, which is the most popular animal model for the Parkinson's disease cell therapy field, we found that these differentiated cells completely rescue the rotation behavior. And then using the humanized mouse, we also found out that when we transplant the patient autologous dopamine cells, they are not rejected at all, as shown here. But the allogenic dopaminergic cells were completely, totally rejected in the humanized mouse model. So then also we developed a novel neurosurgical device where we can generate, instead of the bolus formation, we can generate more columnar, which is increasing the surface to volume ratio, and then also increase the dopaminergic cell survival after transplantation. And there is the apparatus, which is developed by the company, FHC. And then we used this device, and now upgraded to ROSA robot for the future neurosurgery. So based on this progress, we contacted FDA in 2015, and then after submitting many amendments, we finally got the approval for this single patient extended access to treat the first patient, and we got the IRB approval. Right before this, the patient whom we wanted to treat came to me in 2017 in July, and he grasped my hands and told me that, Kwang-Soo, I am dying. His condition was really worsening. He could not swallow, he could not get out of the bed, a very terrible situation. But luckily, one month later, we got the whole approval. And FDA asked us to do the surgery only on one side of the brain because this is the first in human case, and the safety is not proved. They requested us to do the surgery on the one side and wait 6 months. And then if everything is okay, then we can go ahead for the second surgery. So that's what we did. So we did the first surgery in 2017 September, and the second surgery in 2018 March. So this is some snapshot of our clinical trials. The first surgery was performed at the Cornell Medical School, and at that time we used the freshly harvested dopamine cells, and then we delivered using the private jet, and then delivered it to the OR room, and then we did the surgery. Our clinical colleagues did the surgery, and the second surgery was done at the MGH. And we followed the patients using the imaging analysis, such as MRI scan and the PET scan. And as you see here, right after the first surgery, that his brain MRI, the PET scan, dopamine activity was really worsened. But then after the second surgery, the PET scan results showed that the dopaminergic activity became improved and sustained. And then also UPDSR scores and neurological exams show that the patient improved modestly, not drastically. But most important message was that after the 2 years of the follow-up, the patient did not experience any side effect, any adverse effect, including the graft-induced dyskinesia or tumor formation or any other adverse effect was not observed. So we published this first report in NEJM in 2020, and our work was highlighted many journals, including Cell Stem Cell and Nature Medicine. And in 2023, our work was selected as one of the 25 breakthroughs in the stem cell research history. So in summary, can you say that we proved the safety and efficacy of autologous cell therapy? Of course not, with this N of 1 study. However, this N of 1 study is still very meaningful. And during this work, 2 axioms were really encouraging us. The first one was in Talmud, which says, whoever saves one life saves the world entire. And the second axiom is in Bible, which says, what will it profit a man if he gains the whole world and loses his own life? We have to address the safety and efficacy of autologous cell therapy in the next phase 1 clinical trial using with more patients with more comprehensive design after IND enabling CRO and non-CRO studies. And another important aspect is that the cost for this autologous cell therapy is very expensive, and how can you make it cost down? Based on our experience for the human genome sequencing, which started with several $100 million to sequence one person, nowadays it's 1,000 or 2,000. So with these technical advances, we believe that the cost for this autologous cell therapy can be also go down and become affordable for many patients. Using the remaining several minutes, I'd like to share with the more recent result. By the way, to do the FDA approval, we did the IND enabling preclinical study using the GLP and non-GLP study, starting from the 4 patients, and we generated multiple iPS lines and then identified clinical grade iPS lines from each patient, and then we did all the QC tests. And then we also further improved our differentiation method, and now this work is under review. And furthermore, we recently established our own cGMP facility at MGH, and this is some photos, and which is now fully validated and then keep on running. So to do the next stage, phase 1/2 clinical trial for 8 to 10 patients, we contacted the FDA right after the first patient's surgery in 2018. And then they requested the CRO studies as well as non-CRO studies. So we did the biodistribution, tumorigenicity, toxicology test. We submitted our IND application to FDA in 2022, but then they requested a lot of additional data. So we submitted 13 amendments in response to their request. And we got a hold letter in 2022, August. But then we addressed all those issues and submitted complete response. And last year, October, FDA lifted the clinical hold. So we got the full approval. And we registered our clinical trial in the clinicaltrial.gov with this inclusion and exclusion criteria. If you are interested in more detailed information, you can go to this clinicaltrial.gov with the NCT number. So then, after doing the first patient clinical trial, we got many questions come out. So this is now from bedside to bench. And for example, the most compelling question is that why the first patient's recovery was so modest? Because this is the autologous, and we did all the QC, and in animal models, it completely cured the behavior. So we expected much robust recovery, but it was not the case. And so why the recovery of PD patients with the previous fetal cell transplantation was also variable and insignificant? Maybe there is a common underlying fundamental reasons. And so eventually, the question is why the survival of dopaminergic neurons after transplantation is minimal after transplantation. Actually, this issue was the most important, hardest issue when people try to do the fetal cell transplantation, because in those cases, the supply of the material was severely limited. So they studied this issue, the poor survival, very extensively, and there come out many possible reasons, hypotheses explaining. Still, it was not completely explained. So we thought maybe there is an still remaining question, remaining reason for this, which is it possible that the surgical procedure itself could be a risk factor? So we did a series of experiments to address this, and we transplanted the human-derived dopamine cells to the mouse model, NSG, the immune-deficient mouse or humanized mouse, and then after we sacrificed them after 2 weeks, and then analyzed the graft. And then we found that, as you see here, the human nuclei positive cells, which means that the transplanted cells, they survive okay. They survive about 80%. However, when we analyze the transplanted dopaminergic neurons, by the way, when we transplant, the cells have about 15% dopamine neurons, and the remainings are progenitor cells. So among those dopaminergic neurons, after 2 weeks, they almost all died. So you can see the significant difference. So then in the humanized mouse, the peripheral immune cells were infiltrated to the graft size. So we further analyzed, to analyze it, maybe that means that surgical procedure itself could be a risk factor. And so we then, next to perform the experiment, we injected the needle with a buffer, without any cell. So in that case, we found that there is a lot of phenomenon, such as the cytokine expression and the neutrophil infiltration and microglial activation, astrocyte activation, and T cell, B cell. So surprisingly, what we found out is that this phenomenon after the single needle injection was almost identical to the phenomena observed in the traumatic brain injury. So based on that, we named it needle trauma. So as you know, in the traumatic brain injury, a lot of things happen, and eventually regulatory T cells go there and then regain the homeostasis. So we thought that maybe, what if we co-transplant the regulatory T cell from the first? So we did it, and we found out that autologous regulatory T cell almost completely diminished needle trauma. So by all indications. So then finally we did co-transplantation of the autologous regulatory T cell with the dopamine cells. And then we found that in this case, the recovery was more complete, and the recovery happened much faster. So we published this in 2023, and in the News and Views, Professor Qizi Tang at San Francisco nicely summarized our work. So when we transplant the iPS-derived dopamine cell, it triggers the needle trauma, and then lots of the inflammatory molecules are induced. And then also there are some unwanted proliferation, but if we co-transplant Treg cells, it can suppress the inflammation, needle trauma, and then also suppress the unwanted proliferation, resulting in the better clinical outcome. And Dr. Tang also suggested that this idea of the co-transplantation of a regulatory T cell could be also effective in other types of the cell therapy. So we recently reviewed this whole cell therapy field and highlighting our result. And this review highlights the importance of the host immune responses. And as the previous speakers emphasized, I think we have to deal with the host immune response, which is first adaptive immunity. And to address this adaptive immunity and the graft rejection, most popular way is to use the immunosuppression. But it is very exciting to hear from the previous speaker about the hypoimmunogenic or universal stem cell. That is also, I think, in the future, a very exciting field. And then also, as I mentioned, HLA-matched iPSC can also partially alleviate the immune response, but still, in this case, definitely we need the immunosuppression. And in addition, we have to deal with the host innate immune reaction. And we found, as I showed, that the needle injection itself triggers a huge innate immunity. And to address that, we showed that co-transplantation of the autologous Treg cell could be a way, but still it needs further improvement. And also other methodologies can be developed, such as encapsulation and neutralizing antibody and small molecules. So we need still a lot to do. So in summary, again, we established a platform of iPS-based personalized cell therapy for Parkinson's disease. And based on the single patient, we are now launching phase 1-2 clinical study. And human iPS-based autologous cell therapy can become a reality, but I think we need a lot more studies. And we have to deal with both adaptive and innate immune responses. And again, as previous speakers emphasized, we have to think about the pros and cons of the autologous versus allogenic. And I think both approaches should go together. I just summarized our recent reports to deal with the whole process. And I'd like to thank all my lab members, as well as the amazing collaborators, especially clinical collaborators at the MGH and many other collaborators. And thank you for your attention.
Unfortunately, we're running a little late, so I don't think we really have time for many questions. I think we need to move on to the next speaker.
All right.
Thank you again.
Now I'd like to introduce our next speaker, which is Professor Lijian Hui. Professor Hui is located at CAS, the Center for Excellence in Molecular Cell Science. And today he'll be talking to us about cell identity conversion, liver regeneration, and cell therapy.
Okay. First of all, I'd like to thank the organizers for this opportunity to share our recent progress in, I would say, mainly on cell therapy, to fit more the topic of today. Liver is probably the most regenerative organ in the human body. So you can cut liver, 2/3 of the liver, and the rest of the liver will regenerate. In animals, probably in 2 weeks. In humans, a couple months. And even the liver has such a huge regeneration capability. If you injure the liver repeatedly, it will develop all these fibrosis and eventually end to liver failure, end stage of liver failure. And for most of these liver failure, end stage liver failure patients, there's probably no therapeutic curable way except liver organ transplantation. But usually you are lack of organ for such kind of therapy. So in the field, back to many years ago, people proposed cell-based therapies for end-stage liver patients or other genetically related liver disease. One is the so-called artificial liver therapy. So it's basically liver dialysis, I would say. You take let's say you have a liver failure patient, and you take the blood out of the patient and go through a machine, circulation machine, and you put functional hepatocytes in the machine and let the blood go, or I would say plasma go through the functional hepatocytes and go back to the patient in a way to support the patient with some liver functions and let the patient pass through those critical phase of liver failure. And another approach is people were more familiar with hepatocyte transplantation. In this way, you infuse the cell to the diseased liver and hope they will engraft into the liver and repopulate the liver and provide functions. And no matter which approaches, you need a functional, expandable human hepatocytes. And these hepatocytes will take as a medicine for the treatment. But in this case, you really need a huge amount of liver hepatocytes because the liver is big, right? Really big. So to get human hepatocytes, you can isolate from the donor organs, and in this way, you compete with liver organ transplantation. If the surgeons get the organ, they will first use it for transplantation, not for cell therapy, for apparent reasons. And also, very interestingly, hepatocytes, when you isolate these cells from the liver, they put them into petri dish. They don't proliferate. It's a technical hurdle or challenge, I would say, for almost 30 or 40 years. There's no way to expand them in the petri dish, right? So this is the challenges people facing the whole field has to face. So in 2015, Sangeeta Bhatti, she wrote a very nice review to describe all approaches or technical strategies to address those questions, how to get expandable functional hepatocytes. I will not explain all these things to you. Very complicated. People are thinking about many ways to generate functional hepatocytes in culture. I will just focus two approaches which we have generated in the lab. So back to almost 15 years, more than 15 years ago, we managed one approach, so-called transdifferentiation. So we convert fibroblast cells. We can isolate it from the skin or from umbilical cord, whatever fibroblast. When we culture these human fibroblasts, we overexpress three liver-specific transcription factors called FOXA3, HMF-alpha, and HF-alpha using lentivirus. When these transcription factors are overexpressed in fibroblasts, they convert the fibroblasts into hepatocyte-like cells in 2 weeks. Okay, so in this way, you can get hepatocytes independent of the donor organs. And we can expand them by inactivate RB and PP3 pathways so that you get a huge amount of hepatocytes in the lab, functional, expandable hepatocytes in the lab. Along this technique, we actually worked very hard in, I say, 15 years, like to optimize the protocol and try to apply these cells in ball. Why we use it for ball? Because these cells, we inactivate PP3 and RB, so that there's always kind of concern about the safety issue. But if you put it into the ball, it's a device out of the patient. You can take membrane, semi-permeable membrane between the machine and the patient, so it's very safe. You are not you don't worry any about the tumor genetics, anything. So in this way, we first proved the concept using the mouse tail tip fibroblasts to convert it into high hep cells. Then the high hep cells in 2014. Then we demonstrate we can use these cells and put them into ball, and we treat them in mini pig to demonstrate the effects in the large animals. Then we realized we cannot do it all in the lab. Then we transfer the IP to a company. I'm also a founder of the company called Hexcell. Hexcell is hexagon cells, which describe the shape of the hepatocyte. And then they initiated IIT in liver failure patients, which was published nicely last year in Cell Stem Cell. And enabled IND 2022 by CDE in China, and they completed phase 1 now in first half of this year, and they are now initiating the phase 2A trials by Hexcell. So that's briefly the transdifferentiation we have done in the last 15 years. But then I will use the rest of my time to mainly introduce you about another approach in the lab. So we were not that satisfied with transdifferentiation technique because of all these foreign genes you bring to the cells. Because in this case, you cannot transplant them into the patient, I mean, into the liver or whatever ways to bring to the body. And I think like we must find a way to not using these overexpression of foreign genes. And if we think about the liver, it regenerates, right? You cut 2/3 and the rest will regenerate very potentially. So the cells must be able to proliferate. But there's a way how to figure out push them to proliferate. So then we will go back to the literature and look at all the published data and also the data we published. In 2020, we summarized the knowledge about in vivo regeneration after injury. I will focus on injury and the regeneration, not the physiological condition of proliferation. In physiological, the proliferation rate is very low. So after injury, you see there are a lot of proliferation ways, like you can convert from bile duct cell to hepatocyte, which we also published in Cell Stem Cell a long time ago. And you can also have, there are also like axin 2 hepatocytes. Some people demonstrate nowadays probably not right. There are also other ways like hepatocyte proliferation, all these things. There's one thing bring us very interesting. Like there are a type of hepatocyte emerge during the injury. We call them liver progenitor-like cells, which are SOX9 HM4 double positive cells. Because these cells have a lot of progenitor features, so we want to focus more on these cells. So then the first identification of these cells was by Ben Stenger in 2013. I think I should have, okay. So hepatocyte, at that time they found like when you injure the hepatocyte with a chemical called DDC, which causes cholestatic injury, you see SOX9 and HM4 double positive cells. And HM4 is a mature marker for hepatocytes. So all hepatocytes are positive for HM4. Then you see some of these hepatocytes around the periportal area start to express SOX9. SOX9 is a progenitor marker for hepatocyte. During the development, hepatoblast cells, they all express SOX9. And this brings a lot of interest for the lab. They published this, and at that time, actually, because SOX9 also expressed in cholangiocyte, they say it's a transdifferentiation. But later on, because of this publication, many people get very interested into this phenotype. And later on, including our lab, many labs demonstrate it's a dedifferentiation. The first evidence is when you isolate these double positive cells, and you make expression profiling, you realize that they not only express SOX9, they express a lot of genes highly expressed in fetal hepatoblast. The strong overlap of these gene expression. So it's an expression, not one single gene, progenitor gene. It's a whole bunch of progenitor-related genes. So we named this as RG genes as a signature for the study. And another very important evidence is tracing the fate of these cells after the injury. So including our lab and also the data from Max Grompe lab, they demonstrated in a humanized animal. In our lab, we demonstrate we're using this very complicated genetic tool to labeling specifically these SOX9 HM4 double positive cells and ask what they will convert to. So when we label them as a red, IFP positive cells, and after injury, you see they generate into hepatocyte all there. But if you look carefully here, in this corner, this place, you see here, this place, you see a very nice red bile duct structure. So these cells, red cells, the bile duct cells are coming from these hepatocytes. Nicely demonstrate that these SOX9 HM4 double positive cells will give rise to hepatocyte and cholangiocyte, which is a typical definition of progenitor cells. And another very important data from this experiment is when you labeling these double positive cells, you get a single-digit percentage, like 5 or some kind of percentage of the whole population. And eventually, they will give rise 20% of the newly regenerated hepatocyte, which means they have much, much stronger proliferation capability compared to the normal hepatocytes. And also these double pulse cells you can see in the injured human tissues, as I show you something like this PBC, HPV infected tissues or failed tissues. So seems like, also I think recently there's a very nice Nature paper from Ludwig Valier's lab. They're showing using organoids that they are also, these double pulse cells can contribute to the hepatocyte and cholangiocytes in vitro. So all these data give us the idea that maybe we can expand the human mature hepatocytes by differentiate them into a state so-called progenitor-like state. Then they will expand, so that we have a lot of human functional human hepatocyte. So that's the basic idea that we tried many years ago, almost 80 years ago. So we managed actually to using this strategy to expand the human hepatocytes, as I show you here. So in a traditional control medium, there's hardly any proliferative hepatocytes, as demonstrated by BRDU incorporation assay. And using a medium we call the Win3A. So there's a lot of other components in the medium, but Win3A is definitely the most essential component to trigger the proliferation. So with this Win3A medium, you see 20% of the cells are proliferating now. So we name these cells called poly-HH, proliferating human hepatocytes. So now we have the capability to expand them in large quantity. During the publication, it's 10,000-fold expansion. It's not that big, but it's already enough for a lot of experiments if we want to do for transplantation or ex vivo gene therapy, whatever. It's already provided a platform to do all these things. So when they look at these proliferating hepatocytes, so and perform the expression profiling, so you will realize and put them into this PCA analysis. The 2D dimension, like all these, the space indicate how they close to each other. So PHH, primary human hepatocytes, is on the your left corner, and the LPC is there, the blue dot, liver progenitor cells. And you see the poly-HH, it's during the passage, they are like shifting. You focus on PC1, that's a major component, one. So you focus on PC1, they shift from the PHH to LPC, but they never reach to LPC. But this is already, it's a kind of mixed phenotype of these cells. So it's a quite homogeneous cells, I will say. It's not because we have a different population in the culture. So it seems like they have been already switched to progenitor-like, but they are not reached to that space. So as I said, we need expandable functional hepatocytes. During expansion of these cells, they became progenitor-like cells. They actually lose a lot of functions. We have a lot of data to show that. So how can we mature these cells? Because we need a functional hepatocyte. So then a long time ago, we already know if we put primary hepatocytes as organoids, 3D aggregates, they will get mature. They form a lot of structures like I show you there by arrowheads. You see like there's a biocanalicular you see there. And here under the electron microscope, you see more clearly this biocanalicular and a lot of microvilli and enrichment of mitochondria, all these things. These are indication of maturation. We have a lot of other functional data. I don't show you the details. So that we say maybe if we put these poly-HH in 2D culture, convert them into 3D organoids, maybe they get more mature. And this is indeed the case. The first thing is when you put them into the 3D organoids, they stop proliferation. Okay, that's a good indication because stop proliferation, which means probably they get mature. And when you performed all this gene expression assay, you see the blue bar is the primary hepatocytes, and the yellow bar is the 2D culture, the poly-HH. And the red bar is the 3D organoids, the poly-HH formed 3D organoids. And you see here that they in 2D culture, they lose functions, all these albumin, TTR function genes. But in 3D culture, they regain all these functional gene expression. And also, you look at the SOX9 and the CD133 progenitor marks, you see like in 2D culture increase and in 3D they reduced. They are not that perfect as primary hepatitis, but this is the best we have in our hand. And then we try to transplant these cells into the liver. So apparently we success in the animal model, mouse animal model, because the FAH model is the best for transplantation. But if you think about to transplant this to the human or to a liver patient, disease patient, then we realize it's very difficult because in a solid organ transplantation, the new cells come in, just like say this room is full of people. How can we put a new audience inside? We need to remove the older audience out, then we can get the new audience inside. The same thing for the solid organ. So that's the problem, at least for liver, is a big issue. And I don't think we have a good solution for this. I talked to so many people in the field. I don't think they give me clear answers. So we're still working on this technical issue. But what does that mean we have nothing to do for these cells? No. There are another actually approach in the field which we have been ignored for a long time. So that people have already think about it because the transplantation to the liver is very difficult. People think about it. Like we can take primary human hepatocytes, then we enwrap them with hydrogel, alginate hydrogel, so that we can transplant these cells into the peritoneal. And these in a way like they not in the liver but in the belly, so that they can function also in there and make their liver function, all these things. So this is a very cool approach. Actually, Anil Dahawan in King's College Hospital, he has already used this strategy for acute liver failure kids in his hospital. So he isolates hepatocyte and encapsulate with alginate hydrogel, and he put these alginated primary human hepatocyte to this acute liver failure patient. He performed 8 of these transplantation, 4 survived, and 3 bridged to liver transplantation. To him, is very significant improvement because according to their criteria, most of these patients will die. Actually, so we are very excited by learning this publication from his lab, and talked with Anil, and I proposed like maybe we can use our poly-HH. He's very happy because he has always problem to get hepatocyte, as I explained to you very beginning. So now we came to this idea. We can expand the primary human hepatocytes. We let them to form liver organoids. Then we'll enwrap them, encapsulate them with alginate, taking the strategy Anil has done in his hospital, so that we can transplant this EL to the belly of the patient. So we first demonstrate this in the animal. So we take in this experiment we take extended partial ectotomy as a liver failure model. In this experiment we cut 85% of the animal, and most of the animal will die because they lack of liver function. Then we support them with this ELO. As you see from 3 different donors, they can all rescue the animal to 60 or 70% of to survival rate. They will have also control as a primary mouse hepatocyte and control from mesenchymal stromal cell. The mesenchymal stromal cell has no therapeutic effect from our point of view. Now we are teamed up with Changsha in Shanghai, and Anil also joined us together. We are now trying to using this EL initiated IIT in the hospital now. So we have actually three patients, 4 patients are already enrolled. So so far the safety is kind of okay. So I think if we really want to use these cells for clinicals, I think there are a lot of technical challenges. I just list a little bit, some of them. And some of them we have actually addressed more. So now one thing is previously we can passage the cell for 8 or 9 passages. Then they go into so-called senescence or other kind of things. But nowadays, we find a combined chemical medium. We can really expand them into passage, let's say, some of the, so these are the, this one is the way, because we keep the passage. The other two, we didn't complete the passage. So it's to 15, 16, 18 passages. It's like millions folders. I think it's enough. I told my students to stop. I say it doesn't make sense to even expand them. It's good enough, because if you calculate 1 liver to 1 million liver, it's enough for my whole life to see the treatment. I have calculated this, so I say it's not enough, not necessary to calculate. But then we encountered another problem. I don't know if other people have also focused on this. So it's genomic integrity. But after this long-term expansion, it's very important to look at the genome stability of these cells. So we're working on these things. Another thing is cryopreservation of your cells, because we are, I think sometimes you on shelf, which means you don't produce it, then you ship to the hospital second day, right? You produce it, you freeze it, then when it is on demand, you sow your cells, you treat the patient. And this, for organoids, I think is a big challenge, because the size of organoids is usually over hundreds of micrometers, and the penetration of this, to cool them down, to freeze them, you really to handle all this crystallization of ice during the cryopreservation. We realize this. So we using conventional preservation, you see there, all cells die after you sow them. But now we have modified. This is really a collaboration, a team up with people working on this, how to say, like cryopreservation. It's physicists. Okay, they are not biologists, but they know all these things. So we team up with them, a lot of modification, including the CPA, the cryopreservation agent, how to nucleate the crystallize the ice, all these things, and how to calculate all the CMO transfer, all these things. I don't go to the detail, but we manage it nowadays so that we can freeze them and can sow them, and I think we can do all this. And I will stop here, like a lot of collaborators and my team, and also where the money comes from, and also for the audience, young people, if you are interested in cell therapy, you are welcome to apply for postdoc and assistant. Yeah, thank you.
Thank you, Dr. Hui. Do we have time for maybe 1 or 2 brief questions? Do we have any questions?
Hi, Li Jian. Very wonderful talk. I'm curious about the pro-HH. Is it differentiated from mature hepatocyte?
Yeah, yes.
So you use 9 chemical compounds to proliferate the cells.
Yeah.
After such a long-term expansion, can the cells still go mature?
Oh, yes.
Yeah.
So we have, why didn't show the data? This is something we have to always test. If they expand them, they don't get remature, it doesn't make too much sense, right? So every passage we test if they can remature them in 3D organoids. Yeah.
So have you ever analyzed the epigenomic status?
Epigenetic.
Because
epigenetic
usually
we
for stem cell cultivation or for some primary cell culture, the epigenome usually changed.
Yes, epigenetic we not yet checked. We only look at the expression profiles.
Okay.
And nowadays we more focused on the genome integrity. Yeah. So in humans, telomere shortening is a big issue to us.
Okay. Okay. Thank you.
Are there any other questions?
Okay. Okay, if not, we'll thank Dr.
thank you.
Lee again. Okay, and now it's my pleasure to introduce our next speaker, Professor Jiani Xiao. She's a professor at Tsinghua University, and she'll be talking about tissue regeneration by human pluripotent stem cell-derived cardiovascular cells. Dr. Na.
Okay, well, it's my great pleasure, great honor to share some of our results with you here. I have learned so much from those inspiring talks this morning. So in our lab, we focus on using human pluripotent stem cells to differentiate two cardiovascular lineage cells. And we are very interested in differentiation mechanisms. But also, now we think they have a great potential in regenerative medicine. Okay, as we know, cardiovascular disease is the leading cause of adult human, is one of the leading causes of adult human death, and also the risk factor of many other diseases, like we all know, like myocardial infarction, like diabetic foot, stroke, and eye vasculopathy. And in all these diseases, very often start from pathological change of the blood vessels affected by numerous conditions, and then lead to tissue death. So in our lab, we use human pluripotent stem cells to differentiate to like cardiomyocytes, smooth muscle cells, endothelial cells, and also some immune cells, and we use them to try to repair the blood vessels and other cardiovascular tissues. So I was trained as a developmental biologist. So when we start to differentiate these cells, we first went back to the embryo and look at where the cardiovascular system are developed from. So and here we noticed that they are actually from this MESP1 lineage, which are marked in green cells here. So the MESP1 cells will form the heart, the blood vessels, and some blood cells in the embryo. So then several years ago, we generated MESP1 knock-in human PSC, and in that study, we show that it's a very interesting regulation that when we need to activate Wnt signaling to get these MESP1 cells, and then MESP1, which is a transcription factor, it will then inhibit canonical Wnt signaling and activate non-canonical Wnt signaling, and then promote the cardiomyocytes or blood vessel lineage differentiation. And we also show that by transplant MESP1 cells, and it can also do a pretty good job in heart repair of the animal model. For cardiovascular differentiation, many people use this protocol to get a cardiomyocyte at a very high efficiency, and this protocol actually is when you think about it, it's pretty amazing that one can remove insulin from the culture media, And as you know, insulin is very important for cell metabolism. But then if one remove insulin in the culture media, actually can get really efficient mesoderm induction, and the cardiomelcite differentiation can get close to like sometimes more than 90% of cardiomelcite. And then we thought, okay, maybe we can also use similar strategy to get endothelial cells and smooth muscle cells. And actually, indeed, that is the case. And we looked into the mechanism a bit. When we remove insulin, actually, we activated autophagy in the differentiating cells. And this is just to show that the LC3, which is a marker for autophagy, just increase when we remove insulin, but with insulin in the medium, then there's much less LC3, much less autophagy. So this actually have several advantages. One is we can really efficiently eliminate all the pluripotent stem cells because they rely on insulin for their metabolism. And the second is we can quickly get cardiovascular lineage cells. Just to show that with this differentiation protocol, we can pretty easy to get nearly 100% pure endothelial cells, and they have a normal endothelial function, which is a tube formation and ingestion of lipid. And also, we can get a pretty high percentage of, a very high percentage of smooth muscle cells, and they can contract after drug treatment. And also because we can get these cells within 6 to 8 days, and they resemble the EC endothelial cells and smooth muscle cells in the early embryo, so they almost don't express immune antigen genes like B2M or the C2TA, so they are naturally pretty hypoimmunogenic. And then we used them trying to, for some transplantation to treat ischemic disease animal models. For example, the stroke model, the mid-cerebral arterial occlusion. And we can see nicely that after transplantation, the area of infarction really decreased, and the transplanted human endothelial cells, actually they stay as endothelial cell fate and form the blood vessels. Here we can see that this human-specific antibody recognize the CD31 antigen in the transplanted area in the sections here. Similarly, we also tried the critical hind limb ischemia model, and this is a model for diabetic foot. And we can also see that transplanted endothelial cells stay as endothelial fate after transplantation up to a month, we can see, and they also can be detected by the human-specific CD31 antibody. And also nicely rescued the blood flow to the hind limb. One of my colleagues, who is an ophthalmologist, actually become interested in our research. And in his practice, he encountered some conditions that there's currently no cure in the clinic, and that is choroid ischemia in the eye. So this is a picture of the eye, and we can see that so there is a layer of choroid between the RPE layer and the sclera layer. And this choroid layer is mostly blood vessels, and it's very, very important for the eye because it is supplied, because about 85%, around 85% of the blood in the eye actually stored in the choroid. So it is vital for the well-being and survival and of RPE and photoreceptors. This choroid is also derived from MESP1 progenies. So here, as shown in green labeled cells. So in clinic, there's becoming more and more common that the choroid degeneration become a factor of blindness. So, for example, in pathological high myopia, retinitis pigmentosa, and AMD, very often they see that there's a massive, very obvious degeneration of the choroid blood vessels, which you can see by the blanks area here. So in that case, even they perform surgery to correct for example, the retina reattachment, or even to transplant RPE cells if you don't repair the choroid blood vessel, and the patient will still lose sight quickly. So we decided to try whether the endothelial cells can restore choroid blood vessels. And first we generated a model of choroid ischemia, which is, this is used, and we can see that in the model, disease model rat, there is like a black hole in the background, and that's the choroid blood vessel being damaged. So this is quite similar to the human disease. For example, the dry AMD, you can see at the macular region, there's also a black hole, which indicate there is a lack of degeneration of choroid blood vessels. And we tried to transplant, inject just a little bit for our rat model, we just injected like 50,000 cells, and waited for 90 days, 3 months, and we can see that these human cells, we use human CD31 specific antibody to track the transplanted endothelial cells, and you can see these cells labeled by green, nicely incorporated with the blood vessels. And we also trying to explore what happened when the human endothelial cells encountered ischemic choroid. So we dissect some choroid tissue and put our endothelial cells on top of it. So the choroid tissue, as they are all micro blood vessels, they are very sensitive when you take them outside the body. You can see that, and here is the in vivo choroid capillaries. They are kind of fat and fully filled. When we get them outside, they just very quickly become flattened and start to degenerate. Within hours, and when we put the human ES cells on it, we can see that the human ES cell form the blood vessels on top of ischemic choroid. So we checked gene expression change by single-cell RNA-seq, and we can see that there are many genes upregulated in these endothelial cells, like regulation of vasculature development and response to decreased oxygen level, cell junction, cell migration. So not only these cells become more mature within 48 hours, and also they upregulated many genes, and some of them have a neuroprotective effect when they encounter the ischemic choroid. So we look at the fluorescent angiography of the cell-transplanted choroid, and we can notice that this black hole was gradually filled with some blood vessels, and interesting. So this is how we inject. We inject ECs into the supracorridor space, which is it's a potential cavity in between the choroid and the sclera. And we can inject only 1 or 2 microliter of cells, about 50,000 to 10,000, 100,000 cells, which is very little compared to other type of transplantation. And they're not only integrate to the damaged blood vessel, but also they can migrate to other places and also incorporate to both choroid capillary, media vessel, and large vessels in the choroid, as shown by the green staining here. So as we age, actually the choroid in our eye becomes thinner and thinner because the blood vessel there become aged. So when we are about 5 years old, our choroid probably like 200 micron thickness, and by the time we're 50, probably is 20 or much thinner choroid there. So when we look at the histology sections, we find that endothelial cells actually can make the choroid thicker. So, for example, the normal rat choroid is 17 micron, and after transplantation, they become much thicker. And we did some quantification, and it's significantly thicker even than the wild type. Okay, and the most important thing, if this procedure can improve the retina function, so we also test the we also perform the electroretinogram, and indeed the retinal physiological response to light was increased, become more sensitive after endothelial transplantation. So whether this is safe, because eye is so sensitive, and we actually compared with mesenchymal stem cell injection, and if inject umbilical cord mesenchymal stem cells to the supracorridor space, actually they become more proliferative, which will cause edema and inflammation in the eye. And at first, actually, I had some doubt about transplanting endothelial cells to the eye, because it's well known, neovascularization is a problem in AMD and in many eye disease. But my colleague said in neovascularization, those blood vessels, they are abnormal. So when they perform the angiography, they can always see the leakage of the dye from those pathological blood vessels. But in our experiments, actually, we never see any leakage of the dye from the regenerated blood vessels. So that indicates they are safe. So we are quite happy to see all this effect. And we think this might be a potential therapy for the choroid ischemia in the the pathological high myopia in the future, and we are now translating this technology. And also, as I showed you before, so the dry AMD also have a choroid degeneration. So we are now working on whether this can also repair in diabetic animals, repair the choroid in diabetic animals. So this work was recently published. Okay, I'm going to switch a bit to our other research. We can become very interested in tissue residence macrophages because they are important members of the cells in the tissue microenvironment, and they are derived from, for example, from yolk sac or from some from the very early AGM cells, and during embryo development, they just enter all the developing organs and stay as residence macrophages, like Kupffer cells, like microglia, and also in the heart, there's also cardiac residence macrophages. And this is a nice picture from one of my PhD students who working on macrophage differentiation from human pluripotent stem cells. And all these red cells actually are macrophages. You can see there's so many of them in this PM is a novel porous microscaffold material. So recently, we developed a way to using this PMS material to generate lots of macrophages from human pluripotent stem cells. It's actually very robust. So we can see that, and also actually it's very cost effective. We first just induce mesoderm cells and load them into this PMS scaffold, and then first induce endothelial formation, and then add a bit MCSF to induce macrophage differentiation. And then we can get a massive amount of macrophage just flown out from the PMS. So because this is this material can be moved to different places, so we can put them into the bioreactor and scale up macrophage production. So here we can see that this using this protocol, we can also get nearly pretty pure macrophages as shown by flow cytometry, and also the cell number. We can continuously harvest macrophage like for 2 months. And they are similar with macrophages in vivo by gene expression study. One interesting we find is in this culture system, the PMS actually they were start to appear. There's many host cavities start to appear, and then you can see that the round hematopoietic cells just hang in these cavities. So what happened, we find, is differentiating endothelial cells actually well modify this material to generate like blood, to form blood vessels with a diameter about like 50 micron or even bigger, like small blood vessels in the embryo. And these structures are very amenable for macrophage induction. And our gene expression analysis also show there's a very high expression of macrophage self-renew and the proliferation genes. And also there is a population of macrophage that, there's a high percentage of G2M in G2M phase, indicating they are actively proliferating, probably due to there's many pro-proliferation factors within this microenvironment. Okay, so what can we use this macrophage for? actually, this human pluripotent stem cell derived macrophage, they are very active in engulfing, they have very strong engulfing ingestion activities, but they don't produce as high as inflammation cytokines as adult macrophage, peripheral blood macrophages. And here we can see that when we in vitro incubate them with E. coli or this SP bacteria, and they can actively engulf, for E. coli, almost like 100% can engulf the GSP coli, and more than 50 of them can engulf SP bacteria. So, and actually, these pneumococcus bacteria strains are antibiotic resistant, so they are drug resistant. So in human, bacteria very easy become become drug resistant, especially in older people. They have reduced immunity. So, and here we transplanted this IMAX to the pulmonary pneumococcus infection model. So we first infected the mouse with this drug-resistant strain, SP6A, and after about a day, those mice actually are immune deficient. So, so they are pretty seriously affected by this SP strain. And but after we transplant macrophages, and then we can see that really the bacterial count become decreased, and the body weight of these infected animals recovered. So we also look at the lung histology section and find that transplant macrophages can really help to reduce the pathological change in the lung. And one of my colleagues actually who gave us this pneumococcus recently showed that actually liver is the major organ to clear those these bacteria. So we look at the liver too, and find actually there's also the morphology of the liver are much better after macrophage transplantation. Okay, so so this story we are submitting, trying to publish it now. So this is to show that this novel biomaterial can help to produce a large amount of macrophages and for transplantation therapeutic purpose. And we have another story that using these macrophages to generate macrophage-induced cardiac organoid. I think my time is up, but just to quickly go through it, we find that macrophages can really induce, enhance the physiological function of the cardiac organoid. The cardiomyocytes were also derived from human pluripotent stem cells. We can see that it's improved the sarcomere structure, improved the beating function. And in gene expression studies, we show that actually there's the macrophage promoted the cardiomyocyte maturation and the connectivity. But also the cardiac microenvironment also reprogrammed the macrophages to make them become like in vivo cardiac macrophages, which express some myofiber genes. You can see that not only the expression of myofiber genes increased, but we can also detect some muscle proteins in these co-cultured macrophages. And also we did receptor ligand studies and identified some macrophage factors potentially can improve the cardiomyocyte function. And as we tested here, indeed, for example, one of the macrophage factor can significantly improve the calcium transit of cardiomyocyte and the connectivity, which indicate by the CX43 expression and Ki67 expression suggest indicate improved proliferation as well. Our previous study have transplanted those mixed cell type cardiac organoid, which consisted of cardiomyocyte, endothelial cells, and smooth muscle cells, and find this mixed cell type cardiac organoids can repair myocardial infarction much better than the pure cardiomyocytes. So now we are conducting this experiment to see whether with the macrophage infused cardiac organoid, we can also improve the heart repair. So basically, our lab just study how to get those cardiovascular cell types and some immune cell types, and now we are more collaborating with the clinical doctors and tissue engineers and trying to use them for regenerative medicine. So there are some graduating students from our lab, and we have many very great collaborators that help us complete those experiments. Thank you.
Okay, thank you, Dr. Na. We have time for a question or two. Any questions? Okay, perhaps we'll just thank Dr. Na again. And, oh, you guys don't need to do that. We're just kidding. So our next speaker, unfortunately, could not be here today, so we'll just close our session early and head over to lunch. So our afternoon session will begin at 1:30 p.m., so we'll be happy to see you all again at 1:30. Thank you.
And you just talk will be on insulator modified lentiviral globin gene therapy for beta thalassemia.
Okay. Good afternoon, everyone. I'm going to talk about our work on beta thalassemia gene therapy. I'm from Shanghai Jiao Tong University, and also I have the declaration that I am a co-founder of BetaGene. And for beta thalassemia, as you know, this is a very common genetic disease. It is very common in south part of China, also in Southeast Asia and Mediterranean regions. And if we look at the age of those patients, we will find that most of patients are between 6 to 15 years old. This is because this disease is very serious. They are very severe. The patients with this disease, they have a very short lifespan compared to the normal people. That means those patients usually they will die after 20 or their 30s. And the cause for this disease is quite actually it's quite simple. It's because the reduction of beta globin gene. And this is because there's a mutation usually near this gene, and that causes the overproduction of alpha chain. Because the balance is break, then it will produce more alpha globin. Then that is toxic to red blood cells. That means there are two strategies to reverse this disease. One is to upregulate beta globin to make the ratio of alpha globin to beta globin to 1:1. Also, because the gamma globin can also bind to the alpha globin. Because gamma globin they have a very high affinity to actually to alpha globin, and this is used in fetus before baby birth. Currently, there are 4 ways to treat beta thalassemia. One is standard blood transfusion, and also allogeneic HSC transplantation. Then red blood cell maturation agents. Most recently, the gene therapies has been approved. One is Casgevy. The other one is beta cell. Those 2 gene therapies, they use different strategies. One is use CRISPR, the other is use lentiviral vector, and there's different mechanism behind this. For the CRISPR strategy, the gamma globin will be upregulated, and for the lentiviral strategy, the beta globin will be upregulated. Although these 2 gene therapies have been approved, but questions remains. For example, for the CRISPR strategy, as we know, HbF is shut down after birth. In adults, the HbF only exists in a very low level. And why is that? We don't know. And if we turn on HbF again in the adults, what will happen? This is something we need to think about it. In a baby before birth, the baby use HbF to take over oxygen from the mother, because HbF has a higher oxygen affinity to oxygen than the HbA. But if you think, if the mother receives CRISPR gene therapy, then she will also have very high level of HbF. Then what will happen? it might cause some trouble for the baby to take oxygen from the mother. This is something we need to observe for a very long time. And also, evolution choose HbF in adults. Why is that? This is also we don't know. There's much literature about this, the long-term effects of high HbF in the adults. But there's one literature in an island that shows those people with SNP associated with high level of HbF has a high risk of cerebral vascular atrophy. So, if patients receive CRISPR gene therapy, then they get very high level of HbF, much higher than natural condition. Then what will happen to those patients? This is something we also need to be careful and thinking of. And for the lentiviral strategy, there's also some questions that open. For example, we cannot control where the lentivirus inserts. That means if the lentivirus insert at the wrong place, that might activate the oncogene. This is not just in theory, actually. This happens in real clinical trial. At the beginning of 2020, then at the beginning of 20th century, actually, then people use gamma retrovector in the gene therapy, and they find that patients are more easy to get blood cancer. And also, most recently, in a clinical trial that applied run by Bluebird, and they find lentivirus also cause cancer in about 10% of the patients. And the mechanism is the insertion activate the oncogene nearby. So we must be very, very careful when we use those insertional lentiviral vectors. So we are thinking of if there's any very simple way to reduce the insertional mutagenesis. We are particularly interested in insulator because insulator can be very short. So we designed an assay to evaluate different insulators, and we find a very short insulator, which is only about dozens bases. It can very efficiently block the GLP expression. We insert our insulators between the promoter and the GLP, and if the insulator works, then it will block the GLP expression. Then if the insulator is not a real one, then the GLP will be expressed normally. And by doing this, we narrow down to a very short insulator. It's about it's less than 100 bases. So for this insulator, it will not reduce the titer of lentiviral production. And we insert this insulator at the 3 terminal of lentiviral vectors. And during reverse transcription, the 3 terminal of lentiviral vector will be copied to 5' terminal. Then we use this insulator modified lentiviral vector to transduce hematopoietic stem cells. And then, so this is a very traditional way of ex vivo gene therapy. The HSC be modified by lentiviral vector, then it will be cultured in vitro, then transplant back to the patients. So this is how the final product looks like. It's about 20 ml in a small bag, and the liquid looks transparent. And in an IT trial, we transplant these modified lentiviral vector products back into the patients, and we recruit 2 patients with genotype of beta allele. This is the most severe form of beta thalassemia. And for those patients, they received 2 units of blood every like 3 weeks. Then patient 1 received 2.6 times 6 cells per kilogram, and patient 2 received 7.9 cells per kilogram. And they were dosed about 3 years ago. And after transplant to those patients, we analyze how the progenitor cells they produce the daughter cells. We firstly use single-cell DNA sequence to analyze how they change, and before transplantation, it's about 60% cells are transduced by lentivirus. And after transplantation, there's a slight reduction at 6 months for both patients. Then slowly it comes back. And after about 2 years, it recovered to about same percentage as before transplantation. That is about 60% for both patients. And also, we use copy number analysis to follow how those cells, how those insertions changes. And we find that the copy numbers inside transplant cells, they're quite stable. Especially for the patient 2, it's almost a straight line. And for patient 1, there's a slight variation, but it is between 2 and 4 copies. So that means the insertions can be maintained in the transplant cells. And then we look at the proteins produced by the transplant cells. We use HPLC and find that we cannot detect a cell that produce the modified T78Q beta globin at the beginning. Then it gradually grows. Then to about like 6 months, it goes to quite a high level. And after about 1 year, the ratio of alpha globin and beta globin becomes balanced again. It is almost a 1-to-1 ratio. So before treatment, as we showed previously, that the alpha globin is a lot more than beta globin, and that causes imbalance of those globins and cause toxicity to red blood cells. So that's why the patients before transplantation, their blood cells has very short lifespan. And if we look at after transplantations, the lifespan of red blood cells significantly extended. So for the patient 2, it is about like 4 times change, and for patient 1, it's about 2 times change. And we also look at the hemoglobin, the overall hemoglobin level in those 2 patients, and we find that the hemoglobin was above 19 grams per liter for almost 3 years. One is more than 3 years, the other is almost 3 years now. That means the both of these patients are clinically cured. They don't need blood transfusion anymore. And we also look at the neutrophil and the platelet engraftment, and we find the BD211 products have a very short engraft time. Both patients about 16 days for neutrophils engraftment, and on average 22.5 days platelet engraftment, which is much shorter than the commercial products. And for the lentiviral vectors, we are very worried about the insert into the wrong places. So we do sequence on those insertion sites. We find the top insertion sites, they are different at a different time. The drug product means the product before transplantation, and after transplantation, month 3, month 6, month 12, they all have different top 10 insertion sites. And for patient 2, it is almost the same. The top inserts, they are different. That means there's no dominant clone was formed, in our transplant cells. And additionally, we use single-cell RNA sequencing to analyze if the oncogene are activated in those patients. And we find that there's a suspected cancer gene was activated, but then it's not like persist if it disappear later. So that means at the RNA level, the insertion profile are also safe. And more directly, if we look at the height of those patients, they grow quite fast after gene therapy. As you may know, those patients, they are usually very short. They cannot go high because they have trouble in oxygen absorption. For example, for the patient 1, she grew 8 centimeters in 1.5 years, and for the patient 2, she grew 8 centimeters in 1 year. So that is, it's more than they grow in 3 years before the treatment. And this is how they looks like after receiving gene therapy, and both the patients and their parents, they are all very happy. This therapy is free, and they never expect that one day they will be transplantation free. Current days, there's so many genomic engineering tools like CRISPR-based editing and prime editing tools are available. So there's maybe people must think that lentivirus, they are old technology, they are they're too old, they are not fashion anymore. But actually, lentivirus can also keep evolving. For example, we can do surface engineering on lentivirus to make them to touch the hematopoietic stem cells in vivo, and also even the T cells in vivo, to make in vivo products that can significantly cut down the cost of gene therapy, because this is the price is a significant problem for gene therapy products now. Even the products approved, the patients, it is cost too much for the patients, especially for those beta flam family. they usually from very poor families. So it is very important to develop cheaper gene therapies for those patients. And also we can do structural engineering on the lentiviral vector to deliver protein, MI, and IMP instead of viral RNA. And by doing this, we can develop in vivo gene therapy and also super vaccines. And here's an example. We package CRISPR-RNP into the lentiviral-derived VLP, virus-like particle. By doing this, we can shorten the exposure of CRISPR-Cas9 in vivo. That can, on one hand, reduce the off-target cleavage, and on the other hand, may reduce the immune response. And here's the application of this kind of VLP. We are currently doing CRISPR therapy for Huntington's disease, and we have finished a preclinical study, and we are also talking to the doctors and preparing for an IT trial in China. Our strategy to use the lentiviral vector-derived VLP to carry CRISPR. And the CRISPR has two guidelines. It will cut the CAG repeats of the Huntington's patients. And we're also running another clinical trial that is for glaucoma. And this, the first patient has been dosed in August this year. And after about 3 months, 4 months observation, that we find the drug is very well tolerated in patients. It only causes very weak immune response, only a few cytokine upregulated, but not too much. And interestingly, the patients, this patient, he is complete blind, and after receiving therapy, he claimed that he can tell day and night. Before treatment, he cannot distinguish day and night. And we hope this technology can be developed into a next generation beta thalassemia therapy. That means we only need to produce virus-like vectors, so the end product will be VLP instead of cells. That will save a lot of cost, a lot of time, and cut down the price of beta thalassemia gene therapy. So summary, in our study, we treated two beta thalassemia with insulator modified lentiviral gene therapy. And they are completely blood transfusion independent for about 3 years. And secondly, no serious adverse effects was found. No clonal expansion was detected. And thirdly, lentiviral vector and its derived VLP will, I believe, will have a broader application in gene therapy, including HSC gene therapy, even in vivo CAR-T, and also in vivo genomic editing. And lastly, I would like to thank to my team and my collaborators. Thank you.
Thank you very much, Professor Tsai. Any question for Professor? Okay, there's one question.
Thank you so much for your report, and it's very interesting for lentivirus and using in vivo. I think it's very brave. And so my first question is about your lentiviral vector. So I thought you have an intron in your lentivirus vector design, but I know usually when people using lentivirus, they don't include the intron in their target gene. So can I ask why did you put the intron?
Put an intron. Yep. So the intron can make this gene more stable. So this is case by case for the HBB. If we have intron, it will express more HBB protein. And we also tried remove of the intron, and we find the protein expression is much lower than with intron.
Right. And the second question, so for your CRISPR method to treat Huntington's disease. So I know Huntington's got the repeats, but I'm kind of curious, how can you to cut repeat to a normal level, or did you just cut all the repeats? Because I think if you cut all the repeats, it's going to be a problem, isn't it?
Yep. So we cut those repeats by using two guidelines. So we have two guidelines on both sides of the repeats.
Right, but the repeats is they have all the repeat. How can you design a guide RNA can target the amount of repeat that you want to delete?
Yes. So it is the guideline is just before and after CAG repeats.
Right. So you cut all the repeats off.
Cut off. Yeah. Yeah, that's our strategy. Also, there's also another strategy that people use just one guideline, single guideline. And the reason we choose two guidelines is because we think one guideline might produce new repeats. The normal repeats CAG, and for example, if you delete one base, then it will produce a new repeat. So that's why we choose two guidelines to cut out the whole repeats.
Oh, so but if I'm not wrong, so if you cut all the repeats, I think it's gonna be another problem, is it? If you don't have Huntington repeats at all.
It will also, of course, it will also cut a wild type Huntington gene. The difference between wild type and mutant Huntington gene is the number of repeats.
Right. Thank you so much.
Is there any other questions for Professor Tsai? Okay, if not, then thank you very much for your talk. Can I have another applause for Professor Tsai? So our next speaker is Professor Ed Wild. He's the Professor of Neurology at the UCL Institute of Neurology and Associate Director of Huntington Disease Center in the UK. Unfortunately, Ed cannot be here today, so he will join us online. And his talk will be on gene therapy for Huntington's disease, progress and prospects.
Hi, everyone. Good morning. Well, it's good morning for me. I guess it's good afternoon for you. Yes, I'm really sorry not to be able to join you in person. I was delighted and honored to have been invited, but unfortunately, British Airways had a different plan in mind for me. And the aircraft that I was supposed to fly on broke down before takeoff and couldn't be repaired, and BA was unable to supply an alternative flight to get me there on time. In the UK, Friday the 13th is an unlucky day, so maybe the universe always had this plan in mind for me. Anyway, I'm very pleased to be joining you remotely from my home in East London to talk about gene therapies for Huntington's disease. And I was interested to hear the previous discussion. I don't know what happens if you take all of the CAGs out of the Huntington gene and then try and produce a protein from it. But I think what you probably get is something like the form of Huntington found in very primitive species like Dictyostelium and sea urchins or uni, which is probably going to be better than having mutant Huntington around, but a very interesting thought experiment anyway. I'm here to talk about things that are a bit more sort of clinical in Huntington's disease. A couple of disclosures. Essentially, I advise many companies on how to produce drugs to meaningfully deflect the course of Huntington's disease. And I have no personal or financial interest in any individual therapy. I merely hope that we by pursuing many things in parallel, that we will be able to treat Huntington's disease meaningfully sooner rather than later. So a quick word on Huntington's, because I'm aware that this is, unlike many of the meetings where I speak, this is not a dedicated Huntington's disease meeting. So this is one of my patients, Kim, who has Huntington's disease. And you can see that she has involuntary movements, which we call chorea, and loss of voluntary motor control. Kim has relatively early HD because she can still walk. Well, she could at the time of this video. It was a few years ago, and sadly she can't anymore. Each of her children has a 50% risk of having inherited the gene, and the disease is unrelentingly progressive, and there is nothing available so far that has slowed down the disease. But that is the goal. And we're driven by a knowledge of the underlying genetics, which drives knowledge of the pathology. And to put things very simply, nothing stays simple in Huntington's disease for very long, but at the simplest level, each CAG encodes glutamine residue, and the cause of Huntington's disease is too many CAGs in the gene producing too many glutamines in the Huntingtin protein. 36 or more canonically produces mutant Huntingtin protein. It looks like this in cells, it aggregates, it causes dysfunction, it causes transcriptional dysregulation, metabolic dysfunction, many other issues, and eventually leads to the premature death of the neuron. And in the human brain, that's what this looks like. This probably looks like a still image, but when it loops back round to the beginning, you can see that it's an animation. This is 4 years in the life of the brain of a Huntington's disease mutation carrier with no clinical signs or symptoms of the disease. And you can quite clearly see the caudate nucleus here, part of the striatum, undergoing atrophy. I'll just play that again. Here we are, undergoing atrophy. But as the ventricle expands, you can see that actually atrophy is happening everywhere. If you look up here, for instance, at one of the sulci, you can see it widening and the gyri shrinking. So a whole brain disease with a striatal predominance, not a striatum only disease. So this is the sort of simple traditional way of looking at how that gene and that protein cause damage. In the nucleus, the DNA is transcribed to pre-mRNA, which is then spliced. The mRNA is exported and translated to full-length Huntingtin, and it's always been thought that the most toxic fragment, which is this exon 1 fragment, was produced by cleavage of full-length mutant Huntingtin, and it probably still is, but more recently things have got complicated because we've discovered, well, my friend and colleague Jill Bates has discovered this premature termination event which can happen during the transcription of the DNA to RNA. And what this results in is a production of a very short exon 1 only mRNA, which is translated to produce the most toxic fragment, which can enter the nucleus and cause transcriptional dysregulation. And this has become very important in how we think about Huntington's disease and the prospects for therapeutics. So if we just restrict our thinking to modalities aiming to reduce the expression of mutant Huntingtin lowering therapies, we can target many sites in this sequence, but not all of them will have the equal probability of reducing this exon 1 fragment. And to be clear, we don't know for sure that we need to do that, but in our current thinking, that does seem to be a desirable thing to try to do, because if we lower the exon 1 fragment, we'll probably also lower the full-length protein as well through most methods. So starting at the DNA, we can think about gene editing or repression of transcription with zinc finger transcriptional repressors. ASO drugs injected intrathecally, in our case, will target the pre-mRNA in the nucleus. And so those can be intronic or exonic sequences. Small molecule splice modulators are now being developed and tested with the hope that this will upregulate a poison exon, resulting in nonsense-mediated decay of the transcript. If we look at the mature mRNA, that can be targeted in the cytosol with siRNA modalities, either with a sort of naked or conjugated siRNA injected intrathecally, or a gene therapy encoding an siRNA construct. And those could be downstream or could target exon 1. And if successful, that would target anything containing exon 1, namely the full-length RNA and the exon 1 splice variant RNA. And so just to flesh out who's doing what in this space, Takeda has long been working, took over from Shire on a zinc finger transcriptional repressor. The ASOs are probably the most kind of mature of the Huntington lowering therapeutics. Roche and Wave launched programs in quick succession. Vico Therapeutics is a more recent entrant to the ASO scene and has an ASO very interestingly targeting the RNA ortholog of the complement of the CAG repeat itself, which is an interesting strategy. Skyhawk and PTC Therapeutics are both in human trials of splice modulators. None of these are anywhere near exon 1, unfortunately, because of the lack of a suitable splice site to be targeted by a small molecule. So these will all target the full-length transcript only. Alnylam very, very recently entered the market a month ago. Well, I say entered the market. At UCL, we were proud to have given the first dose of their siRNA, conjugated siRNA, lipid conjugated siRNA to a human HD patient. But the focus of my talk will be the first gene therapy program for Huntington's disease, which is run by Unicure. So let's set the scene first with a bit of background on what happened when we first started trying to lower Huntington in a targeted way. And the outcome of the tominersen generation HD1 phase 3 trial is probably well known, but and it's depressing to revisit, but we do it anyway, because that's the only way we learn and improve. And so this was a 2-year trial testing 2 monthly and 4 monthly doses of 120 milligrams of tominersen versus placebo intrathecally. And regressively, everything ended up much worse than we would have hoped. In particular, the 2 monthly high frequency dosing patients ended up on this clinical score, which we call the CUHDRS, or Composite Unified Huntington's Disease Rating Scale, much worse than, well, significantly worse than placebo-treated patients. And the 4 monthly patients had the point estimate on the unfavorable side of placebo. This was accompanied bottom left by this marked sort of spike in NFL, which happened early in the first 6 months of treatment and then gradually receded. But it turns out that neurofilament light release into CSF was indeed a predictor of trouble to come and foretold this unfavorable outcome. So when that trial was terminated prematurely in 2021, there was a quest to try and figure out what had gone wrong and whether there was a path ahead for this drug in particular and for Huntington lowering in general. And one of the striking findings was that if you divided the cohort of 800 people into 4 according to age and CAG repeat length, essentially, the patients who were on high frequency treatment still did worse than placebo, but the low frequency patients ended up on the favorable side of placebo. This is a post hoc analysis, so no statistical or clinical significance is claimed, but it was certainly very interesting that these young patients earlier in the disease, perhaps with more resistance to tominersen overexposure, which I think is what we think was the issue, seemed to be on their way to deriving benefit. And interestingly, if you divide all of the patients in the program by exposure, i.e. measured drug concentration, the patients with the low exposure quartile ended up with significant Huntington lowering here in dark red. The NFL spike here in pink was minimal to 0, but there was actually a very early reduction in tau protein, which is thought to be more rapidly turned over than neurofilament and therefore might be an early predictor of subsequent benefit. This was taken to mean that it was an exposure-related issue and that potential benefit could be derived from giving lower doses of tominersen, avoiding some of the inflammatory effects of the ASO backbone. That drug is now in a phase 2 dose-finding study. But this has left us with many questions in the field. First of all, was part of the tominersen failure because it didn't target an exon 1 sequence? Do we need to do that, or is it possible to produce therapeutic benefit without doing that? What about the mutant versus wild type issue? Are we reducing the therapeutic benefit or even producing harm if we lower both alleles in parallel, or should we be striving to target just the mutant allele? It's much, much more difficult to do, but it's possible. What about anatomy? These ASOs are good at targeting the cortex, but not great at targeting the striatum. Was that part of the issue? No one knows how much Huntington lowering, if we can do it cleanly without off-target effects, will be necessary to produce therapeutic benefit. And for people like me especially, how do biomarkers help us? And we can measure mutant Huntington in CSF. We can measure things like neurofilament as markers of potential target engagement. How can we leverage those to make better decisions and bring drugs to the market? One of these questions, I think, has been at least provisionally answered. The PTC therapeutics have a drug called PTC518 or Votoplam, a small molecule splicing modulator. And we know that this is brain penetrant, so it's probably dosing every cell in the body approximately equally with full brain penetration. And so they're actually lowering at 12 months the mutant Huntington protein in CSF by about 40 to 50%. And they have no neurofilament spike whatsoever at 12 months. And this is compared to a big spike at roughly 6 months that was seen with tominersen. And to my mind, this gives us one certain piece of information, which is a very rare commodity in this field, which is that lowering wild-type Huntington by 40 to 50% does not inevitably produce damage to neurons. And so this is kind of a clean slate for the idea of allele non-selective Huntington lowering, because basically it looks like targeting the mutant only is not necessary to prevent the NFL spike that turned out to be a predictor of poor outcomes. So this sets the scene and brings us on to the Unicure program. And Unicure, the therapeutic, I'm just going to call it AMT-130 because, like all gene therapies, the name is unpronounceable. It is an AAV5 capsid. It encodes an siRNA, which targets a sequence within exon 1 of the Huntington gene. So if this hits its target, it will produce sustained, hopefully lifelong reduction in transfected cells of the exon 1 sequence, therefore should suppress the aberrant exon 1 fragment and production of full-length Huntington. It's administered in lengthy stereotactic neurosurgical procedures under MRI guidance with convection-enhanced delivery. These procedures typically take between 12 and 20 hours on the ClearPoint platform. The target is striatal with 2 sites in the putamen and 1 in the caudate. And you can see here some still images of the drug being given to a human Huntington's disease patient into the putamen here. And it's given with gadolinium contrast so that the injection of gene therapy can be directly visualized. And 2 doses, low and high dose, both relatively high in brain gene therapy terms, 10 to the 12 and 10 to the 13 genomes are being tested. So the program is divided into 2 arms, thanks to the wisdom of the regulators. So in the USA, the FDA declared sham surgery to be mandatory. So these patients have an anesthetic of undefined duration and scalpel cuts into the scalp, and then holes drilled into the outer table of the skull only, and then are woken up. In Europe, the EMA and the other European regulators declared sham surgery to be unethical and therefore mandated an open label trial. And this is just one of many cross-cultural, geographic, political really issues that make testing gene therapies in the CNS extremely challenging. Nonetheless, in the US, 26 patients were enrolled, and initially 10 controls of whom about 60% were able to roll over to open label treatment. But interestingly, in the 12-month sham surgery period, a substantial minority of patients progressed in their Huntington's disease to the extent that they couldn't be dosed with the gene therapy 12 months later, which is really probably an argument for not using sham, because those patients underwent anesthetics and surgery and then ended up being ineligible for active treatment. In Europe, 13 patients have been enrolled so far, or rather data have been declared on that number of patients, roughly equally low and high. The program began in June 2020, just at the beginning of the COVID pandemic. A year later, the first high-dose patient was treated. Europe began in February 2022. Later that year, there was a clinical hold for safety, which I'll touch on, and the latest public release of results was in the summer a few months ago, and very, very hot off the press, red hot, 3 days ago, the company gave a very interesting and encouraging regulatory update. So the first update we got from this program was about the concentration of mutant huntingtin in CSF, and this was declared to be promising. But in fact, in subsequent updates, this apparent reduction in mutant huntingtin has not been detectable in CSF. And I suspect that this was really just data from a small number of people that was rather noisy. And in fact, all this represents is random noise crossing the zero line in both cases, because these in subsequent time points certainly have regressed to the mean. It's slightly frustrating, but if you look at the whole volume, the volume of whole brain, the striatum is about 1% of the brain by mass or volume. And so it's not that surprising that, you could potentially target the whole striatum, fully suppress huntingtin production, and it would only produce a 1% signal in the CSF. Essentially, we collectively, and the sponsor specifically, no longer consider mutant huntingtin in CSF to be a biomarker of interest for this particular program, although it is, of course, important for other programs which are targeting whole brain or much larger volumes of brain. The other sort of speed bump that came up was this inflammation story, which started to emerge after the high-dose cohort began to be treated. There were 4 serious adverse events in that cohort, characterized as CNS inflammation, sometimes with increased CSF pressure. Most of them resolved without intervention, with just conservative management. Some did require steroid treatment, usually short-term, occasionally more prolonged. Interestingly, the time course was typically 2 to 3 weeks after the surgery for the peak of this inflammation. There was a lot of edema around the catheter sites. Those scans are not public, but I hope you can imagine what that might look like on a T2 sequence. As a result of that, at the discretion of the treating team, steroid treatment around the perioperative period was introduced. And now there's another cohort underway in Europe and the US testing this potent immunosuppression regime around the perioperative period to see whether that makes a difference. My experience, I had one patient who had this complication, and interestingly, he developed a headache on day 10, was admitted for observation. We did a lumbar puncture and the pressure was mildly elevated to about 30 centimeters, but it resolved. In fact, it seems that the lumbar puncture was therapeutic in that case, and the inflammation resolved without long-term sequelae. And he's my only patient who's ever gone back to work. He'd had to retire medically, and now he's back at work. And maybe that's placebo effect. He's about 2 years into treatment, but it certainly is a very unusual thing to happen to a Huntington's disease patient. So this is what the clinical update started to look like once they started to come in. You can see that the green line, which is the sham-treated patient, stops abruptly at 12 months, and we'll never have data beyond 12 months. So we need a plan B for how to assess these patients clinically. Initially, a simple natural history comparator was used without much in the way of statistical adjustment, simply looking at the trajectories in roughly equivalent patients. Don't read too much into this figure because the numbers are very small and the duration is also small. The subsequent updates, and I'll focus now on the most recent update, tell I think a very interesting story. And under new management, there was a lot of change at the senior levels of Unicure, and I think the approach that's been adopted by the new team certainly has the potential to generate much more reliable and informative insights. I think the crucial difference with the way that the data are viewed now is that we do still use this natural history comparator, but it's drawn from two very large studies, TRACK-HD and TRACK-ON. That was one. PREDICT-HD was the other. Those were natural history studies with MRI scanning, and 154 of those patients match well the patients in the Unicure program, and propensity score weighting was used to adjust, in which you take a large group of real-world patients, some of whom are a good match for the trial patients, others of whom are not, and then you statistically weight them so that the patients who are similar end up contributing more to the signal, but all of the real-world patients you've included contribute something to the signal. And what you end up with is a kind of a weighted set of natural history controls who are the best possible reflection for the patients in the trial. And this was the big update in the summer of this year. We're looking at the CUHDRS, the composite UHDRS, which is a measure of functional, motor and cognitive scores. The external controls are in grey, and at the 24-month time point with 21 patients dosed, we can see that certainly it does appear that the lines are stacking up in the order that we would like for therapeutic effect, with low dose having some improvement and high dose having substantial improvement over the natural history controls. And really 2 years of stability with little to no decline on the CUHDRS is not something that we would expect to see in the natural history of HD. This is what it looks like in aggregate. Low dose has about a 30% reduction, not significant, but the high dose seems to have reduced progression in this data set by 80%. It's very unlikely we'll achieve that in the real world or in the longer term data set, but it certainly is a nice point to be starting off from, and that does appear to be nominally significant. If we look at the components of that, there's a lot of variability, as you would expect when you look at the individual components of a composite. But I think crucially, interestingly, this symbol digit modality test, which is a cortically predominant motor and cognitive score, is the one that has the biggest difference. And it's thought, it's speculated that this may be because the high dose gene therapy has a better prospect of transneuronal spread to the cortex, but this remains to be seen. Where things get really interesting is when we look at neurofilament, and this is just a study of mine, the HD-CSF study, which looked at 2-year interval repeat CSF collection, baseline to follow-up in patients with HD and controls. And in 76% of patients, 2 years progression of HD produced an increase in neurofilament level. And on average, in early manifest HD, which is where the Unicure trial population also happens to be, the increase is about 10 to 15% per year. And that's a fairly reliable, consistent increase. A group of more than 3 or 4 Huntington's disease patients is extremely unlikely to experience a decrement in neurofilament. Now, when we look at the data from the program, we see this large expected peak in neurofilament, which is a result of the neurosurgery and possibly contributed to by the neuroinflammation that we talked about earlier. You can see, though, that it trends back down to baseline in about 12 months in the low dose cohort. In the high dose cohort, as you might expect, it takes longer to settle down, and that probably is an inflammation thing. But tantalizingly, it seems to be sort of grazing the baseline by 24 months. And if we zoom in, you can see that actually both of the point estimates are below baseline at 24 months. If you pool them and compare them to this expected natural history increase, what you see is a substantial difference. It's the difference between up and down. And with neurofilament, broadly speaking, up is bad and down is good. And so this is from the movie No Country for Old Men. I won't go into it in detail, but it's a great film. If this isn't evidence of disease modification, in my opinion, it will certainly do until evidence of disease modification gets here. Which brings us to the latest update from 3 days ago. This program has RMAT designation, which essentially gives priority access to the FDA for novel therapeutics in or untreatable indications. And so it was a type B meeting with the biologics division of the agency. And this was a big update. that I think has widespread consequences for the whole field. The FDA has agreed that an external natural history control can be used as a primary basis for accelerated approval. And in this case, avoiding the need for an additional pre-submission study. So essentially, on the basis of this phase 1/2 trial, fully recruited now, all that uniQure has to do is show that the current trends continue, and at some point, hopefully in the near future, the drug will be licensed, which could potentially reduce the time to market by up to 5 years. Much more widespread ramifications for the field was that the composite UHDRS, which the FDA had not previously been keen on, preferring the individual functional scores, can be used as an intermediate clinical endpoint. And this results in power calculations that reduce sample size for clinical trials by about 60%. And finally, neurofilament light, a molecule dear to my heart, may serve as supportive evidence of therapeutic benefit. And I think this is a really solid and wise decision by the FDA, which will allow us to run much quicker and more convincing trials of drugs that are hopefully going to protect neurons. And it also gives us a way to bridge from manifest to pre-manifest patients with ISS stages 0 and 1, in whom suppression of neurofilament alone may be sufficient as part of a package to demonstrate that the drug works across the disease. And so this was the image that ChatGPT came up with to represent how I'm feeling right now. I do feel like with this FDA update, Christmas has come early for the Huntington's disease community. I'm not sure I'd want Santa Claus to operate on my brain, certainly not with a household drill, but you get the general sense that there's great optimism. We don't know whether the current trends will continue. It's really interesting to see whether the use of immunosuppression might be able to reduce the size and width of that NFL peak. The third point is kind of a joke, but also kind of not. Hopefully the FDA will still exist in a year, but it might have a very different mindset. And we can't predict the future. Certainly, no one can predict the political future of the United States. So there are challenges ahead. And even if everything goes really well, you still have to scale a CNS gene therapy to the global market with all of the challenges that will bring. So a quick look to the future. This is the field as it stands at the moment. Takeda appears to have stepped back, and they've also cut ties with Wave. So the future of the Wave program is sort of in the balance. But there are always newcomers to the field, and it's an accelerating field with many, many players coming. Two newcomers to the gene therapy in Huntington's field, Spark Therapeutics, which has a proprietary AAV capsid. They recently published some of their preclinical data supporting the announcement of a human trial, which they hope to bring to patients in 2025. It doesn't seem to be targeting a sequence in exon 1, so it's a bit of a gamble, but it's intraparenchymal, and so it may be that we can achieve better brain penetration with the capsid, but we may not be targeting the splice variant. Lattice Bio has not declared where their target is within the Huntington gene, but they've released some very early data showing that their sort of evolved capsids may have much better not only local spread, but also transneuronal spread to the cortex. And that brings us to the end. That's my overview of the Huntington's disease universe. This is an image of the Andromeda galaxy taken from my balcony here in London using my telescope, which you can kind of see there. And the message here is that it takes 2.5 million years for the light from our closest galactic neighbor to reach us. But in order to complete the journey and produce this satisfying image, the light has to leave the galaxy in the first place. So a journey of 2.5 million light years begins with a single step. And it feels like we're finally starting now to capture the light of a potentially brighter future, thanks to the work of many people who laid the foundations years ago. And this is a work in progress, but we will not pause until Huntington's disease is a treatable disorder. So thank you for your attention to my talk, and perhaps we have time for one or two questions.
Yeah, we have time for one quick question if there's any burning questions. Yes.
Thank you, Edward. I have a short question. Do you think it's safe to knock down or knock out wild type gene? And also, I have a second question. Do you think it is sufficient to knock down at RNA level? Because there's a preprint paper says that DNA repeats also play a role in Huntington's disease. So do you think just reduce the RNA level, is it enough to reduce the symptoms of Huntington's?
So on the first question, the wild-type Huntington issue is an open question. And there are certainly people who will say that reducing wild-type Huntington at all is too much of a risk, and that it may worsen the progression of Huntington's disease. My view has always been that we already know what happens if we do nothing, and it looks terrible, and everyone dies. Everyone gets Huntington's disease and dies. So we have to do something. And since allele selective lowering is extremely difficult to accomplish, it makes sense to pursue both approaches in parallel. Many people said when the tominersen trial result was negative, many people said that was because of wild type lowering, and that was certainly a possibility. Although I think most of us who are a bit more familiar with ASOs understood that this backbone inflammatory effect was something that could be expected. I think that question is answered now, honestly, with the results of the PTC Therapeutics trial over 12 months. 40% reduction in mutant and wild-type Huntington did not produce an increase in neurofilament or any other signs of harm or toxicity. And so I do think that certainly over the space of at least a year, it's likely that reducing both alleles in parallel is going to produce substantially more good than harm. Although the good remains to be seen, what we have seen is an absence of harm, and I think that represents significant progress. I'm not sure I fully understood your second question, but were you asking about somatic instability and the tendency of CAGs to expand in the brain, and whether that poses an issue for lowering the protein?
Not just RNA or protein.
Oh, I see. Well, yes. I mean, certainly it's distinctly possible that non-protein pathologies contribute to the pathogenesis of Huntington's disease. So direct toxicity of DNA, toxic RNA foci, RAN translation, and this non-canonical transcript all pose potential issues and challenges. What's fairly clear, though, is that consistently reduction of production of the Huntington protein per se, without removing the DNA and through whichever means the protein has been reduced, tends to consistently produce phenotypic benefits in reliable mouse models of Huntington's disease and across multiple models of the disease. So I think most people at this point, even people with a strong view about what is the most important toxic species, most people at this point would have to kind of, I think, accept that it's complicated, and we should try to do whatever we can therapeutically, but that the toxic protein appears to be a target with huge potential that has not yet been fully exploited in humans, and that if one can choose, then targeting the exon 1 fragment or splice variant appears to be the one with the highest therapeutic potential, particularly as doing so will also target full-length Huntington. So I'm completely open-minded and agnostic about all of this stuff. I mean, it's certainly the case that the gene is the cause of the disease. And so we really shouldn't stop anywhere between the gene and the disease. We need to look at that whole space. At the moment, these things are expensive and time-consuming. And so I think rightly, lowering the production of the protein has been the focus. The big question in the field is which protein. And then, but I think we should pursue all therapeutic angles.
Thank you. Could we have another applaud for Professor Wild?
Thank you very much. I hope that the rest of your meeting is a huge success, and I hope to perhaps join you another time.
Thank you. Our next speaker will be Professor Laurent Soubise, Professor Pediatric Neuromuscular Disease at the Neuromuscular Center, Department of Pediatric, University of Oxford in the UK.
Thank you so much. And I don't want to beat a dead horse for Professor White, but you've missed a wonderful welcome and a wonderful organization, and I would really like to thank the organizer for that. I was in a movie mood when I prepared this talk, so I decided to call it The Good, the Bad, and the Ugly. And I realized how different this talk is of the other in terms of being mostly clinical and being not, slightly less positive. Because what I'm going to try to share with you is all my doubt and my questions after using gene therapy in my field for nearly 10 years now. So yeah, the good, the bad, and the ugly. Well, I've got my fingers in many pies. Look at all this conflict of interest. Okay, the good. Well, if I have to pick up the example of the good and my experience, I would say, well, definitely the pre-symptomatic trials in spinal muscular atrophy. Those have been published in Nature Medicine some years ago. Just to remind you, spinal muscular atrophy is an autosomal recessive disorder. It affects 1 out of 14,000 individuals. And in the most severe form, the patients with spinal muscular atrophy just die before the age of 1 year, right? And so it's related to a monogenic disorder. It's just the lack of SMN protein with a null mutation in the SMN1 gene. And just to give you an idea of what we're talking about, this little gentleman here presents with spinal muscular atrophy type 1, so the most severe form. He's super lucky because he's born in northern Belgium, but he's from northern Belgium, but he's born in southern Belgium where we had newborn screening. And he could be identified at birth with spinal muscular atrophy, and he could be treated at the age of 42 days, which is quite late, but he was the first patient treated by gene therapy at birth in Europe. So we were still, trying our best to go fast, even if it's not that fast, 42 days, we can do much better today. Most of our patients are treated at the age of 15, 16 days. And as you can see, is, well, doing well, but probably is already has the very first symptoms of the disease, with a kid who is actually slightly hypotonic at the lower limb. This patient, in absence of treatment, will die in a couple of months, right? And if you wait for the patients to be diagnosed by symptoms and to treat him after the symptoms, then he will get probably wheelchair bound for the rest of his life. But he could be treated, and he became a neurologist. So that's now how he examined his patient. He is very careful about taking the right tool and taking the reflex. And as you can see, gene therapy has restored the reflex of the patient, but he needs to think about taking both sides. Yes, thank you for that. And nevertheless, he is still a little bit symptomatic, right? Look the way he climbs stairs. That's certainly not entirely normal. But the good news is that he could then be included in next generation trial. And I'm not going to disclose it, which one, because it's still an ongoing trial. But just to show, well, he's slightly hyperkinetic, and that's how he now, leaves the clinic. Where is he going to go? I don't know. As far as he wants, probably he will go up to Moscow and then turn left to Kamchatka or right to Hong Kong. I don't know. But he is very hyperkinetic and moves a lot and doing quite well. So these patients with spinal muscular atrophy identified by newborn screening or because they were siblings the outcome today is to figure out if they are normal or not. We're not talking about if they are living or not. We're not talking if they have symptoms or not. We're discussing if they belong to the gray area of being normal or not. So that's, for me, the best example of a success and a good aspect of the gene therapy. The good, the bad. Well, for me, the bad is mostly illustrated by the phase 1 trial that claim efficacy when they are not powered for efficacy. Let me show you another patient. This is a patient with Duchenne muscular dystrophy, and that's this patient 6 months after treatment and before treatment. So I'm going to try to put the two videos simultaneously. Okay, so you can see that this patient is now doing much, much better. He has still a little bit of proximal deficit, but the way he climbs stairs is much faster. He doesn't need the banister. So what is this treatment? What is this, gene therapy or cell therapy? What do you think? Gene therapy, cell therapy? Well, it's actually steroids, right? We just gave steroid to this patient, and steroid is the standard of care for Duchenne muscular dystrophy because there is inflammation. Guys, keep in mind that when we give an AAV, we also give steroids, right? There are 4 different products that are now studied in gene therapy for Duchenne, and dystrophin is a giant, right? It's a big, big gene. So you cannot just put this giant into a teeny AAV. You need to actually have a micro version of the gene that we call microdystrophin, right? And microdystrophin just get the essential of dystrophin and is now packaged in different types of AAV under the control of different promoters. Well, the first thing to keep in mind is that microdystrophin is microdystrophin, and dystrophin is expressed in a standard form for millennium by all possible animal species, which means that Mother Nature never thought about it before we did, which is not a very good sign. Nevertheless, phase 1 trial published in JAMA Neurology, and as you can see, after the gene therapy, amazing restoration of the muscle tissue, and from a clinical perspective, probably you don't know this scale, it's the North Star. Some patients improve by 11 points on the North Star. And North Star is a score up to 34. When you improve by 11 points, it's massive, right? Even a blind medical student can see it. It's really a massive improvement of the patients, 11 points on the phase 1 trial. But when it comes to a double-blind, randomized placebo-controlled study phase 2 trial, then there is no difference anymore between placebo and treated patients. And the difference is actually, and the improvement is 2 points. We're not talking about 11 points anymore. We're just talking about 2 points. And when we go to a phase 3 trial, double-blind, randomized, placebo-controlled study, then the difference is 0.6. So we're not talking about 11 points. Keep in mind that these patients, on top of receiving the gene therapy, received high dose of steroids, right? This drug was approved. This drug was approved in the US. It's not yet approved in Europe. It's not yet approved in the UK. I just come back from Egypt and from Romania, because every year I go to see patients in Egypt and in Romania. I can tell you that nearly all parents ask me the same question. How can I get access to gene therapy? They don't care about now steroids, about standard of care, about physiotherapy, because they truly believe that now there is a cure, and that the only question is, how can I get access to this cure? Even if I have to take the terrible decision for my family, like relocating in bad condition, how can I get access to this cure? To a cure that fails the phase 3 trial. So, you know what I mean. I think that from a US perspective, from a European perspective, sometimes to approve drugs on the basis of a negative trial can seem to be acceptable because we feel that there is an acceptable benefit to risk ratio, that we need to incentive the field. But we need to appreciate that what we are doing in the US, in Europe, in Asia, has global consequences on the patients, because patients now are connected on social network. And for a family living in Egypt with Duchenne muscular dystrophy, it's the triple jeopardy. You have Duchenne muscular dystrophy. You know there is a cure for your son, but you cannot get access to it because you are living where you're living. And the triple jeopardy is to make the wrong decision and to relocate in bad conditions. And this is not only about this specific gene therapy. If you take another one, which is the product from Pfizer, phase 1 trial, amazing results. Look, when we plot them on a natural history study, and if we conduct exactly what we've heard, a propensity-matched analysis, it's p-value of 0.001, but when it comes to a double-blind randomized placebo-controlled study, game over, there is no difference. And one of the obvious reasons is, of course, the exposure to steroids, that you will never find in a natural history study patients who all of a sudden receive 1 mg per kg of steroids. So I think that we have collectively a responsibility to look at phase 1 trial and say, guys, this is an amazing phase 1 trial. And a phase 1 trial aims to ask the question of the safety and the feasibility and PK/PD, but they are not powered for efficacy. They are not designed for efficacy. And probably we as investigators, the sponsors, the reviewer of the paper, the editor of the paper, but also the readers of the paper, people listening the talk, we should collectively take the responsibility to remind that a phase 1 trial is a phase 1 trial. And now the ugly. The ugly is a little bit all of us. And that's what I was saying, pharma investigator, investors, patients, advocacy, regulators, editors, reviewer. When we collectively generate unrealistic expectations in the community. And I think that it's an important point, because gene therapy is considered as a cure in the image, but it's not a cure. I mean, even my patients with spinal muscular atrophy, identified at birth and treated at birth, and who are running now in the stairs, I don't call them cured. I call them treated. I call them doing amazingly well. But I have no idea what's going to happen in 5 years or 10 years. Are they going to continue being that well? I don't know. This is the son of my grandson who will call it a cure, but not me and not my students, right? So the problem is that understanding and presenting gene therapy as a cure give the impression that one shot and that's it. So it means that if I have a child with a condition and I can get access to a gene therapy, I can concentrate my effort on that, and that's it. And of course, it gives rise to crowdfunding. And believe me, over the last 10 years, I've seen many, many patients coming to me after crowdfunding. Because if you live in a country where you have no access to innovative therapies, you can appreciate that if you can get this $2 million, this $3 million to get the fix of gene therapy, then you have a chance to be cured, and that you don't need even standard of care afterwards. And some years ago in Belgium, in my little country, we had not at that point the gene therapy for spinal muscular atrophy. We had the oligoantisense nucleotide, and patients were treated by oligoantisense nucleotide that works as well as gene therapy for SMA, and a family was smart enough to get $2 million in 3 days of crowdfunding. 3 days, can you imagine? Belgium is a small country, so to get $2 million in 3 days, you have to be smart. You have to be well connected. You need a good network. You need to know how to speak, right? When to cry, when to laugh. But these guys were smart. They could get access to gene therapy to get an add-on treatment of oligoantisense nucleotide, which may or may not make sense. But it was presented in the newspaper as a massive achievement. This family, because they were fighting, could get 2 million to cure their child. And then my patients came, my patients next door. They came to me, they say, "Doctor, look at that, there is a cure, I want this cure." And they started crowdfunding themselves. But as you might imagine, people are ready to give for one child, for 2 children, but not for every single children. So the second, the third, and the fourth one, they don't have the same success. They organized amazing dinner during which they could raise 500, 600 euros, but they were still missing 1,900,099,400 euros, right? And for these parents, that was the triple jeopardy. They have a patient with spinal muscular atrophy. They are told there is a cure, but they cannot get the cure, and they cannot get the cure because they're not smart enough. They are not good enough. They are not like the other parents who could organize the crowdfunding and get the magic fix. So we are actually generating wrong expectation, and we are placing the fault and the guiltiness on the shoulders of parents who have already a child who is affected by a severe condition. So we have discussed in some papers this issue related with crowdfunding, and I'm not going to detail all of this. I'm just making some self-promotion for our work. But just the cost of crowdfunding is huge. There are privacy issues, consent issues, ethical issues for donors, but also, and that's for me the most important, equality. I mean, if you are white, if you are middle class, you are much more likely to be successful in your crowdfunding campaign in Europe than if you're not. And that's exactly the contrary of social security. Social security is a system in which the state and the citizens collectively give an equal access to treatment. And crowdfunding is placing the children on the marketplace, and to give people the opportunity to put their thumbs up or down and to select those that they want to treat. And of course, if mom is pretty, if mom knows when to cry, when to laugh, how to present things, they have a better chance. But is it the rationale on which we want to treat patients today? Is it to have a real and well-organized social security, or to have a Hunger Games in which patients are selected on the basis of an equal one? If you don't like crowdfunding, I've got a bad news, there is worse. There is the lottery, right? And after the first crowdfunding campaign, Novartis understood that it was quite difficult to deal with that, and organized and made a wonderful gift to the field. They offer 100 fix for free of gene therapy, and this fix of gene therapy were available on the basis of a lottery. So which means that if you live anywhere in the world and you have your son with this terrible spinal muscular atrophy, you can apply to gene therapy, and every 2 weeks there is a lottery, and you can have the gold ticket, get the gold ticket, and be treated by gene therapy for free. But as you might imagine, this gold ticket does not include the standard of care. The gold ticket does not include the fact to be treated in a center with good experience of gene therapy. So at the end of the day, is this gold ticket quite a gold ticket? Well, unfortunately, gene therapy was applied sometimes in very poor conditions and led to the death of at least 2 kids. So we need to figure out that this type of issues, that is crowdfunding and lottery, comes from the wrong expectations that we have collectively raised in the field. And for me, this is one of the example. This is the initial paper in the New England Journal of Medicine for spinal muscular atrophy. And as you can see, these patients on the shopping tent, which is a 64-point scale, are improving. But obviously, 2 patients are doing extremely well, and these are these 2 patients who raise immediately the floor of the scale. And there are videos. I had video in my initial presentation, but then I decided not to display it. But of these patients doing amazingly well, they are normal. And nobody has never seen a patient like this with spinal muscular atrophy type 1 treated by gene therapy. And the reason is that these two patients were identified by screening. But it's not a problem. It just means that when you treat early, these patients are doing amazingly well. But if we present these patients asymptomatic, and we show and we display video of patients normal, if I'm the dad, if I'm the mom of a patient with spinal muscular atrophy, I would say I want this drug, I want this cure for my kid. And as I said, because of this lottery, the gene therapy was delivered in several countries, and unfortunately, as I say, 2 children died. And then it brings me to the issue of the risk perception. This is a patient of mine who actually drawn the logo of my team, and I had a discussion with him. Say, let's consider that I have a very efficient drug. It can cure the patient, but I have 10% of casualty, 10% of death. My patients could think, did I say something wrong? So these patients will say, with this drug, I have 90% of chance to go better. And I have 100 patients, so if I use this drug, I will kill 10 of my patients. Patients think individually. We think collectively. And so the perception of the risk is very different. And I appreciate that definitely I apply the risk to my entire population. A patient has a risk of, is a binary issue, right? Is it going to be a fatal risk or not? And if not, I can manage. So we need to appreciate that, and to be honest, my views have changed over the time, and probably I appreciate that, especially when it comes to a very severe condition, you can probably change your mind, but again, we need to properly inform our patients, because the right decision that parents take for their children is when they love them, but when they are properly informed, and when they're not properly informed, they don't take the right decision. So the take-home message of my talk about gene therapy in neuromuscular disorders is that, yes, we have achieved a tremendous success, and we can be collectively proud of that, and we can admire people who have been the pioneer for that. It has demonstrated the potential to give a normal life to kids who would otherwise die or survive with severe disability, like my little neurologist. But there are important ethical questions that go far beyond formal GCP. It's not just ticking boxes or explaining a consent. It is really considering what we are collectively doing when we present our data, when we read this data, when we review this data, when we publish this data. And we really need to adapt our ethical standards to this new reality of a world that is global and to which we present this data. And with that, I would like to thank my team in Belgium and in the UK. Thank you so much for your attention.
Thank you so much, Professor Servais. Very intriguing, thought-provoking talk. Is there any question for Professor Servais?
Thank you very much for your very inspiring talk. I cannot agree with you anymore about your suggestions to consider, about the ethical parts and safety parts. Even in my experience, after our first patient case, I got hundreds of the emails and phone calls still until now from all over the world. But my impression by listening to your talk and the previous talk is that these patients are really desperate, and we got to do something. We all agree with that. But at the same time, the potential negative impact of our trials a little bit in a rush without securing the safety completely, I think we should very seriously consider. For example, I hope to ask questions from the previous speaker. For example, Huntington's disease, for one example. It seems like we still don't understand very fundamental biologics. And I don't know about your diseases, but for example, even Huntington disease, I think I can ask Dr. Kai, that I remember there has been a dispute about whether Huntington disease is a loss of function disease or gain of function disease. So there have been a dispute. I don't know whether there is a clear conclusion. So even without that kind of a fundamental and basic mechanisms about the disease, if we hurry on the therapeutic intervention, I think is it really do more harm or more gain? So that was kind of echoing your concerns. And if you have further thoughts.
Thank you so much. And if you give me 30 minutes more, I can answer this question. Long story short, I fully agree. I think that we need also to have a disease-dependent approach. When it comes to, well, I try to put myself in the shoes of a patient. If I'm diagnosed with ALS or Huntington, I think that my level of risk acceptance will be quite okay, right? But when I speak about a Duchenne patient with 3 or 4 years old today and who has a life expectancy of 30 years, that's pretty different, right? Just for spinal muscular atrophy, if I speak about SMA type 1, in which patients will die within 5 months as a baby, I think that the risk should not be understood the same way than if you have a SMA type 3 with ambulant and doing in the community. So there is, for me, just the first question is the question of the trajectory, right? And we need really to understand, and I think that the risk acceptance should probably be different. Regarding the understanding of the biology, I think that for SMA and Duchenne and the other conditions that we are considering, we have quite a good understanding. But sometimes there is something that we have missed. And for me, the best example being X-linked myotubular myopathy, which is a terrible condition in which male because it's X-linked, they don't move at all, even not the eyes, right? They don't move at all. And it's an enzyme, myotubularin, right? So the gene therapy is probably the most efficient treatment I've ever seen in my bloody life. You give gene therapy to these patients, some of them could be went off ventilator and walking. But in 23 patients injected, 4 died because they were also a liver toxicity that is really disease specific. What means safety? What means risk in a condition where you don't move at all? I don't know. And I think that the question is also cultural. We know that the risk understanding, for instance, in the UK or in the US is massively different, right? US has no problem with risk. UK is a little bit more, right? But we really have to understand that also in a disease that we believe we know, sometimes there is some hidden part that makes our life more difficult than we initially believed.
Thank you very much. Can we have another applause for Professor Survey? So our next speaker is Professor Alexandra Biffi, Professor of Pediatrics at the University of Padua, but she unfortunately cannot join us today in person, so she has kindly recorded a presentation that we will play for you now.
So good afternoon. It's a pleasure to be here with you today. I apologize again for not being able to be there in person, and I thank the organizers for the kind invitation. So in the next 25 minutes, we will discuss together about the use of hematopoietic stem cell-based therapeutic approaches for the treatment of neurometabolic and neurodegenerative disorders. I would start just describing to you the platform that we use, which is extremely versatile and that was developed for originally the treatment of primary immunodeficiencies, hemoglobinopathies, so blood disorders. The platform is based on the use of autologous hematopoietic stem cells that are collected through standard apheretic procedures and are then genetically modified by the use of lentiviral vectors or other gene therapy platforms in order to correct the patient's genetic defect in the case of monogenic diseases. The cells, after that, are infused back into the patients, usually by intravenous delivery, and usually after the administration of a chemotherapy that favors the engraftment of the transplanted cells. This process has the ability of progressively reconstituting all blood elements with transplant-derived cells, and this is why it could be beneficial for blood diseases. However, the same process can also reconstitute extravascular hematopoietic cells, such as macrophages, dendritic cells, and importantly for us today, also microglia cells residing in the central nervous system. Indeed, the transplant progeny cells in proper transplant conditions have the ability to generate a myeloid progeny that will progressively acquire microglia functional features. And these cells could have a huge therapeutic potential for the treatment of neurometabolic and neurodegenerative diseases. First of all, because they could restore the scavenging and homeostatic functions of microglia that is metabolically incompetent or activated in the diseased brain. But they could also modulate and reduce tissue damage and neuroinflammation, and eventually, thanks to gene transfer, they could become able to generate a durable source of therapeutic molecules across the blood-brain barrier, such as in the case of lysosomal enzyme in lysosomal storage diseases. This concept has been proven effective and true and real and beneficial in neurometabolic diseases, not only in animal models, but most importantly in patients. And in two conditions, metachromatic leukodystrophy and adrenal leukodystrophy, the concept has been so advanced to lead to the registration of two drugs that are based, in both cases, on the use of autologous hematopoietic stem cells and lentiviral vectors that encode for the disease-causing gene. In both conditions, one is a lysosomal storage disease with very severe neurodegenerative features. The other is a paroxysomal disorders, again, very severely affecting the central nervous system of the affected children. The treatment in both has clearly demonstrated not only to compensate the gene defect and restore metabolic functions, but also to provide significant therapeutic benefit to the affected children. And this is why, clearly, the drugs are now available on the market. In the case of MLD, both in Europe and in US and in UK, for XALD, only in US. Similar data are accumulating in a few other indications that have already reached clinical testing stage, such as mucopolysaccharidosis type 1, and also in our lab we have other applications of the same approach that are in development, the most advanced of which is in the setting of mucopolysaccharidosis type 2, also known as Hunter disease. I would just spend a couple of slides on this just to tell you that Hunter disease is clearly a lysosomal storage disease with unmet medical need, specifically for those patients who are characterized by central nervous system involvement. These are children who develop pretty early in their life cognitive impairment and developmental delay because of the inherited defect in the activity of the iduronate sulfatase enzyme. And these children cannot benefit from enzyme replacement therapy because the recombinant enzyme cannot cross the blood-brain barrier. Well, in this case, we think that our HSC gene therapy approach could be particularly beneficial, and because of that, we worked from preclinical model up to clinical readiness in this indication. And in this slide, you can see what constitutes the basis of our research in terms of proof of concept of efficacy that we obtained in the animal model of the disease using a lentiviral vector encoding for a codon-optimized human IDS cDNA and applying the same treatment paradigm I presented to you before. Well, in the animal model of the disease, HSC gene therapy resulted efficacious not only in preventing disease manifestation when treated pre-symptomatic animals, but most importantly, in correcting disease manifestation upon treatment of symptomatic mice. And this was shown in terms of behavioral abnormalities, skeletal abnormalities, histopathology, and biochemistry. All this was achieved in the absence of safety issues or adverse events, and the clinical translation of these findings were further supported by additional safety data that were generated in ad hoc models of dedicated safety testing. Based on this data, as I told you, we are now close to clinical testing. We completed CMC activities. We have GMP batches available for clinical testing. We built a clinical protocol with the help of an international clinical advisory board, and we are now fundraising for launching, hopefully soon, a phase 1/2 clinical trial to treat hunter children. So you would say at this point that so far so good, we are in a good spot, but I would say up to a certain point. And the reason for that is because all this that I told you, which is a clearly proven evidence of benefit associated to the use of HSC gene therapy for treating CNS affecting neurometabolic diseases, is limited in terms of applications to patients who are pre-symptomatic at time of treatment, or who have very limited central nervous system involvement, who retain a quite good function of their central nervous system. And the reason for this, that is intended obviously at maximizing clinical benefit, is dependent on the very slow pace that the process of microglia reconstitution following HSC gene therapy takes. Indeed, this process would take in humans something between 6 and 12 months to happen. And during this time, before the engraftment of the transplant-derived cells in the central nervous system is enough to provide benefit, the disease per se could continue to progress. And this is clearly a limitation. Actually, in my laboratory, we have been working a lot in order to address this limitation. And in particular, we are trying to develop advances in terms of technological ameliorations of the treatment paradigm of HSC gene therapy that could allow us to address the complexity of neurometabolic, but eventually also of neurodegenerative diseases with rapid progression, and eventually for already symptomatic patients by HSC gene therapy. And the way by which we are trying to do so is based on 3 key elements, the most important of which is the opportunity and the possibility to anticipate timing and extent of engineered cell engraftment in the brain, obviously to benefit symptomatic patients and rapidly progressive disease. To this goal, we have worked on 2 modalities, on 2 approaches. The first of which was based on a very, very simple observation that we did in the lab several years ago. In particular, what we discovered is that the transplant-derived cells in the brain are not only derived from a progressive process of monocyte infiltration from the peripheral blood into the CNS, process that would mostly lead to perivascular macrophages, but is rather dependent by an early seeding of the central nervous system by a fraction of the transplanted hematopoietic stem cells that through the circulation would reach the CNS, engraft locally, expand locally, divide, and generate a microglia-like progeny. So based on this hypothesis, we propose to modify the standard administration route of hematopoietic stem cells, that is intravenous, and to actually deliver hematopoietic stem cells also in the central nervous system, either intracerebroventricularly in the lateral ventricles of the brain, that is important for our discussion today, or intrathecally. And what we have discovered by mimicking this alternative route of administration in preclinical models in mice by using differentially labeled hematopoietic stem cells is that when combined to standard intravenous delivery of hematopoietic stem cells, the intra-CNS delivery of the same cells in the brain lateral ventricles increases significantly the contribution of the transplant to microgliosis and favors the degeneration of a mature microglia-like progeny cells, as we have shown through multiple assays looking at the transplant-derived cells gene expression dataset, but also morphology and phenotype. So we decided to exploit this finding and to develop an innovative HSC gene therapy application for a very severe and very rapidly progressive disease affecting the central nervous system. This disease is a neurometabolic lysosomal storage disease called CLN1. It's one of the ultra-rare neuronal ceroidal lipofuscinosis due to the defective activity of lysosomal enzyme PPT1 and characterized by severe, massive, rapidly progressive neuronal loss and neuronal degeneration. We decided to address the complexity of this disease exploiting a standard knockout animal model, mouse model, and we applied to the PPT1 knockout mice the standard treatment paradigm I described thus far to you. So transplantation of hematopoietic stem cells from knockout donors transduced with lentiviral vectors encoding for the PPT1 cDNA into myeloablative transplant recipients. However, in this case, we modified our original recipe and we either transplanted the cells intravenously or we transplanted them only in the lateral ventricles of the brain or by a combination of the two approaches. And the transplantation was performed either in pre-symptomatic or in already symptomatic animals. And to make the story short, we could demonstrate that by employing this strategy, we could restore PPT1 activity in the brain of the treated animals, and we could provide therapeutic benefit, both in terms of behavioral abnormalities correction, in terms of histopathological alterations, and clinical features. Importantly, however, what we could clearly show that at every measure that we evaluated, and both in the pre-symptomatic or symptomatic setting for treatment, the treatment modality that outperformed over the others had been the combinatorial approach. So whenever we transplanted the gene-corrected cells, both intraventricularly and as standard intravenously, the outcome had been the better, in particular in terms of behavioral correction, histopathology, and biochemistry. This obviously to us makes a strong rationale for proposing for clinical development HSC gene therapy in CLN1 disease, and in particular to propose to the affected patients the combinatorial delivery of the gene-corrected hematopoietic stem cells, particularly whenever the patients are symptomatic at time of treatment. And in order to actually be able to do so and to deliver this concept into the clinics, also in this case, we accumulated safety, feasibility data in dedicated preclinical models. We completed CMC activities, and also in this case, once completed a clinical protocol definition with an ad hoc clinical advisory board, we started fundraising that is still ongoing. And we really hope to accomplish with that in order to be able to open a phase 1-2 clinical trial as soon as possible. Importantly, if we go back to the original model I proposed to you for the intraventricular delivery of hematopoietic stem cells, we also discovered something else. And in particular, what we understood is that if we just track and follow the hematopoietic stem cells that had been administered in the brain and their progeny, we would end up understanding that this could result in a successful, rapid, and CNS-restricted engraftment of transplant-derived cells. So the cells that we transplant in the brain not only reconstitute CNS microglia, but also achieve reconstitution exclusively in the brain. They are not able to engraft in the marrow. They remain locally. And sincerely, we think this could be a very powerful way to approach not necessarily only neurometabolic diseases, but also adult onset neurodegenerative conditions for which clearly CNS targeting could be extremely relevant. And in this direction, I will give you a couple of examples, the first of which is in the area of progranulin deficiencies. For those of you who have some neurology background, well, you would know about the many roles that progranulin exerts in the central nervous system. It is a secreted glycosylated protein that is usually produced by neurons and microglia that has a number of very relevant neurotrophic, anti-inflammatory, and lysosomal functions. Importantly, whenever progranulin is lacking in the central nervous system, a sustained pro-inflammatory environment is established, neurodegeneration occurs, caspase activation occurs, neurons would die, lysosomal dysfunctions would appear, and oxidative stress as well would happen. And this translates actually in two disease conditions. On one side, we have CLN11, another steroidal lipofuscinosis that is due to biallelic GRN mutation. It's an ultra-rare disease characterized by a very, very severe and rapidly progressive neurodegeneration and neurocognitive decline, lacking curative treatments. On the other side, we have the GRN-associated forms of frontotemporal dementia that are forms of FTD that are caused or that are associated to monogenic mutations, monoallelic mutations in the progranulin encoding gene. Actually, these cases are not rare. Up to 5% of frontotemporal disease patients would carry haploinsufficiency at the progranulin locus. So this is a condition that could account for a huge number of patients because of its high penetrance. And we can consider that up to 2,500 GRN FTD prevalent patients at the moment exist in US and in Europe per year. So clearly, you would agree with me that targeting GRN deficiency could be very relevant because of its large epidemiological impact. And we decided to address it by exploiting our treatment paradigm, so transplantation of hematopoietic stem cells that are genetically modified in order to express the progranulin under the control of a standard ubiquitous promoter. The possibility that this treatment paradigm could be efficacious and potentially valuable, not only for CLN11, but also for frontotemporal disease, was assessed in a relevant disease animal model, which is the GRN knockout mouse. And in this model, in which we applied the standard treatment paradigm that, again, I described to you many times, we challenge the system by introducing an alternative vector with a linear specific promoter of our invention, and by introducing the delivery of the genetically corrected stem cells only in the lateral ventricles of the brain, instead that intravenously. Well, what we could demonstrate in the GRN knockout mouse is that whatever the combination of these factors was, we could achieve therapeutic benefit. In particular, if you look at this behavioral data, at this biochemical data, either in terms of markers of accumulation of undegraded lipids, or in terms of, again, accumulation of lipofuscin in the brain of control and treated animals, you would agree with me that the phenotype that is very evident in the knockout untreated or more treated mice is reverted to the wild-type phenotype upon treatment with either the PGK or the HLA lentiviral vector, with either intravenous or intracerebroventricular delivery of the gene-corrected cells. And this is true also for other disease manifestations that relate to neuroinflammation, microglial activation, and astrocytosis, telling us not only that HSC gene therapy could be a valuable therapeutic opportunity for CLN11, but also progranulin-dependent frontotemporal dementia forms, but also that in these settings, a CNS-restricted approach based on administration of the gene-corrected cells directly in the central nervous system could be efficacious, with a huge advantage in terms of safety and feasibility of clinical translation. Very similar considerations can be done if we look at another key player in CNS neuropathology, that is TREM2. TREM2 is a triggering receptor expressed on myeloid cells. Particularly, it is expressed by microglia. it's a key molecule in the CNS because it possesses the ability to modulate critical cellular functions via crosstalk with diverse signaling pathways. And whenever TREM2 is lacking, microglia function is compromised. And in particular, this results in alteration of immune responses, in phagocytosis, in cytokine production, in microglia proliferation, and microglia survival, leading to neurodegenerative manifestations. Well, also in this case, we have diseases specifically linked to TREM2 deficiency. In particular, we could have B-allelic TREM2 mutations that cause an ultra-rare autosomal recessive inherited disease called nasoacular disease that presents as a progressive presenile dementia with onset in the 3rd decade of life that usually leads to death in the 5th decade of life. It is a real microgliopathy. It is characterized by microglial dysfunction. And on the other case, we have monoallelic TREM2 mutations that predispose to Alzheimer's disease. Indeed, TREM2 is one of the highest threats, along with APO2 variants, for increasing the risk of developing Alzheimer's disease. And in particular, several rare variants of TREM2 have been linked to an increased susceptibility to Alzheimer's disease in humans. And in particular, the variant R47H is the most common Alzheimer's disease genetic risk factor. This why? Because actually microglia, as you know, is highly involved in Alzheimer's disease pathogenesis. In particular, microglia cells, thanks to TREM2 surface receptors, are able to not only contribute to eliminating and consuming beta amyloid plaques, but they also are involved in the removal of neurofibrillary tangles and tau deposits. So they have a key role that is strongly related to TREM2. So based on these findings, we hypothesize that HSC gene therapy could be beneficial also for nasoacular disease and TREM2-associated forms of Alzheimer's disease, and eventually Alzheimer's disease in general, because the mechanism of microglia removal of debris and of amyloid and of tau remains valid also for those patients who are not defective in TREM2 expression. So based on this, we again work in preclinical models of the disease. We started working on a very classical and very penetrant and severe model of Alzheimer's disease, which is the 5X FAD mouse that carries 5 different mutations that in humans account for an increased risk of Alzheimer's, but not TREM2. And we applied to this mouse our conventional approach. We use a lentiviral vector encoding TREM2, in this case a standard ubiquitous promoter, to transduce hematopoietic stem cells from 5X FAD donors, and the cells after transduction were transplanted into pre-symptomatic 5X FAD recipients, either intravenously or in the lateral ventricles of the brain. And actually, in this therapeutic setting, we could again demonstrate the efficacy of HSC gene therapy in mitigating significantly the disease manifestations of Alzheimer's disease in these mice that are reported here as abnormalities in standard behavioral testing, such as the Morris-Water maze testing, and importantly, not only behavioral correction, but also correction of the stigmata of the disease in terms of clearance of beta amyloid accumulation that was observed upon treatment of the FAD mice. Amyloid removal was really possible thanks to the interaction of the human TREM2 expressing cells with beta amyloid plaques, and also correction of other disease stigmata such as neuroinflammation, microgliosis, and astrocytosis. Therapeutic benefit of HSC gene therapy delivered either by intravenous administration or intracerebroventricular administration of the gene-corrected hematopoietic stem cells was not only proven in this animal model, but also in an alternative model that is very relevant also for nasoacular disease, that is the TREM2 knockout mouse. Also in this case, the same treatment paradigm resulted in therapeutic benefit. Here we are looking at two different behavioral testings that again testify and confirm that in the treated and engrafted mice, the behavior that is abnormal in the knockouts is restored to a normal phenotype thanks to the reconstitution of TREM2 activity. So these findings that we consider proof of concept for translational development of HSC gene therapy for TREM2 dependent Alzheimer's disease forms and nasoacular disease are now being developed through a translational plan that is summarized into this slide, and that really is intended at developing a treatment modality for patients through modulation of our treatment protocol, attempting to reduce the intensity of the conditioning regimen for favoring clinical translation, execution of safety and IMPD enabling preclinical studies, and completing CMC and regulatory activities in order to be able to launch a clinical testing plan at the end of the project that has been recently founded by the European Innovation Council. My last few minutes will be dedicated to just mentioning to you the alternative approach or potentially complementary approach we are developing in order again to enhance the contribution of hematopoietic stem cell transplantation to microgliosis. This modality is fancy because it's really based on a serendipity. By studying the mechanism of microglia reconstitution in hematopoietic stem cell transplantation models, we have had the chance to work on a reporter mouse where microglia cells are labeled by GFP through the modulation of the CYP3CR1 locus. For those of you who are familiar with microglia cells, it would be clear that CYP3CR1 is really the microglia signature gene, is a gene highly and robustly expressed by microglia since their origin throughout adulthood in physiological conditions and in pathology. Actually, what we discovered by using this CYP3CR1 reporter mice in transplantation is that whenever CYP3CR1 is defective, such as in haploinsufficiency within the hematopoietic cells, actually it results in a process of favored microglia reconstitution. In particular, when we transplanted hematopoietic stem cells from CYP3CR1 haploinsufficient or knockout mice, these cells were able to reconstitute CNS microglia much faster than wild-type hematopoietic stem cells. And the process was not only faster, but more efficacious in terms of acquisition of morphological and functional microglia features. Well, we decided that this mechanism could be exploited, in particular by a platform that could allow us, thanks to the use of gene editing and targeted addition, to achieve therapeutic gene targeted addition at the CYP3CR1 locus of hematopoietic stem cells from patients affected by neurometabolic or neurodegenerative disease, with the intent of correcting their gene defect by an approach that could allow us not only anticipating CNS myeloid engraftment and time of benefit, because by editing and targeted addition, we would obviously induce haploinsufficiency at our CYP3CR1 locus, but also provide robust and therapeutic microglia-specific expression of the gene of interest at the CYP3CR1 locus of the HSCs and of their progeny, while at the same time potentially reducing safety concerns related to LV integration into the stem cell genome. This concept has been recently described in the paper that you can see here in the bottom of the slide, and is being developed towards clinical translation in the setting of the two neurometabolic diseases we commented on at the beginning of my talk, metachromatic leukodystrophy and adrenal leukodystrophy, in the context of a grant that was recently sponsored by the European Research Council. So I will close here by thanking you for your attention and by acknowledging all of the contributors to this work in my lab and among collaborators. Thank you again, and I would be more than happy to take your questions.
Just learned that Professor Biffy has actually managed to get online. So if there's any burning question, I can take one. Right, if not, then we can break for tea. But before that, can I ask all the speakers and our organizing colleagues from Tsinghua and CellPress to come to the stage for another group photo, please. And from the audience, if you want to join the photo as well, you're more than welcome.
Welcome back, everyone. Let's continue our following session. I'm Jiani Xiao. I'm scientific editor at Med, the flagship medical journal published by CellPress. So it's my great honor to chair the second half of the gene therapy session. So in this part, we will have another two presentations about technical advances in gene therapy, and then we will have 20 minutes for each presentation. After all the sessions, we will move on to a panel discussion. So during the panel discussion, we will invite all the speakers again up to the stage for any further discussions. So let me introduce the following speaker. The following speaker is Professor Wensheng Wei. So Professor Wei is currently a professor at the Biomedical Pioneering Innovation Center, Peking Tsinghua Center for Life Science, and director of the Genome Editing Research Center at Peking University, and also lead scientist at Tamping Laboratory. So today, his topic is about from gene editing to new frontiers, advancing cell and gene therapy. Let's welcome.
All right, thank you very much for the kind introduction, and also thanks for the organizing committee to give me this great opportunity. So I'm going to, hopefully this is going to work. No. This is my disclosure. I'm a founder and a scientific advisor for two companies. So for today's topic, I'm going to emphasize the breakthrough in cell and gene therapy really rely on either we have the original target identification or deep understanding of important mechanism, or we have an innovative way or technology to deal with some of the very difficult or challenging tasks. So I'll give you both directions examples rather quickly for 20 minutes. So for the technology-wise, I'll just give you two directions of cases. One is the LEAP technology we developed a few years ago, which is RNA editing technology relying on native mechanism instead of using foreign proteins and also totally independent of CRISPR system. And we have upgraded the version 2.0, a version of the LEAPer. And I'll give you a few examples how we deal with the mitochondria-based editing and how we prepare for this model for future therapeutic purpose. Again, come back to the LEAPer technology. This is a kind of discovery or invention by accident. We initially play with CRISPR system, Cas13, and accidentally identify and realize if we introduce long enough antisense RNA, of course we have to engineer AC mismatch, we can effectively recruit native ADA for program editing on the A to I kind of conversion. And obviously, we can only need to introduce a very simple and short antisense RNA, make it a very light kind of system. It's friendly for delivery, and also it's great for the safety profile. And we continue to upgrade this very technology for two kind of directions. One is we want to make it as powerful, as efficient as possible. So we try multiple ways to optimize this very technology. And ultimately, we decide to use the circular ARNA. We call ARNA, which is short for ADA recruiting RNA, instead of linear ARNA, whether we can circularize in vitro, actually ex vivo, deliver into the cell, or we can express within the cell as a linear form and circularize within the cell. Either way, we can observe elevates the increased efficiency of editing in most cases, not all cases, okay? But same time, we observed in almost all cases, the prolonged editing outcomes. The reason is because the circular RNA is just more stable compared with the linear counterpart. And we have a very special bystander off-target associated with this very technology because we have to introduce long ARNA. So we will have like Watson-Crick duplex of RNA, ARNA versus the target RNA. So although AC mismatch is a preferred conformation for this target A conversion, which is a diminution rather, but we have multiple AU
perfect matching when you purposely recruit ADA to the duplex of duplex double-stranded RNA, you'll have a chance to have this AU matching adenosine be deaminated, okay? Although we have tried different kind of way to mitigate this very unwanted issue, especially if we engineer the G opposite to A, we can observe great reduction, but we cannot completely eradicate this kind of unwanted editing. So the solution is very straightforward and simple. My student test if we purposely delete the U opposite to this A site, we can almost completely eradicate this unwanted editing. So now we have a kind of a very decent 2.0 version of LEAPR. So if we want to apply this very technology in therapeutic framework, first of all, the advantage or superiority of RNA editing is the safety profile, okay? But the limitation actually is kind of transient, especially if you deliver the RNA translate into the cell, okay? They're only going to last for a short while, whether it's a week or 3 weeks, it's won't be a lasting kind of effect. So obviously, one choice for therapeutic purpose is to combine this with AAV system. So the first thing we tested actually is to use AAV deliver the LEAPR, which is ARNA. And in NHP study, so you can see we can observe up to 13 weeks of a high level of RNA editing. And we predict we can safely see much longer effect of RNA editing, but we have to stop the experiment to collect the data. So in real therapeutic practice, which is the DMD, Duchenne muscular dystrophy, I don't think I have to go into any of the introduction. We have a prior talk on this very topic. So the very problem associated with DMD is this is a protein called dystrophin, okay? And dystrophin is important to collect the intracellular skeleton and actually the matrix to ensure the integrity of the muscle cell, basically. So the defects have a very severe consequence, okay? And this is symptoms you can pretty often observe in male patients. The reason we call male patients is because this is a problem. This very gene is X chromosome linked, okay? So the frequency is pretty high, 1 after 3,500, even to 3,000, okay? And the problem for this very protein coding gene is a huge gene, okay? Including the intron, it takes over 16 hours for one round of transcription, okay? So the problem is often occur in late axon with this very coding gene. So good news for this very problem is 80% of patients, if we can skip the very problematic axon, you might have a chance to restore the reading frame, and consequently, you can have the restoration of the protein expression and also its function, okay? So this gives us the opportunity to try using LEAPR, which is RNA editing technology, to skip axon to restore the problem, okay? So, this is a little, in details for the so-called mechanism, how the LEAPR can successfully skip axon. At the very beginning, we thought it's very straightforward. If we, because we know the SA side, which is the AG side, if we convert A to I, we have a chance to skip the downstream exon, okay? But it turned out, actually, there's a collectively effect of 3 different kind of mechanism by using circular ARNA to targeting SA site. Actually, the RNA editing is only one, is one kind of a contributor for exon skipping. Without further going into any of the details, I just want to show we have collaborated which make this NHP model, which has the deletion 2 nucleotide, or 2 base rather, or 5 base deletion in exon 5, which causes the reading frame shift, okay? So our task by trying this new therapeutic, practice is to skip the exon 5, okay? So this basically shows it's not that straightforward to have a best efficiency in terms of skipping exon using RNA editing. you really have to try multiple sites, in addition to the assay site for the RNA editing. So at the end, we can achieve very high efficiency in terms of exon skipping using RNA editing. So the delivery is the key, which have different kind of version of AAV, but ultimately we choose to use the mono AAV, which initially published in Cell paper 2021. And you can see clearly, if you compare this very delivery system with the AAV9, we receive much decent efficiency for this very ARNA delivery into the muscle cell. And you can see the consequence of exon skipping in the muscle cells. So I will quickly jump into the results, basically. And probably you guys are all familiar with the CK, which is creatine kinase, which is a kinase present within the muscle cell. Because of the inflammation, because of the leaky problem due to DMD symptoms, these very kinds will go into the PBMC. Okay, so this become very sensitive marker to show the severity based on the level of this CK dose. Okay, so you can see the quick and drastic reduction of CK after the treatment, and lasts for 50 weeks until 50 weeks, which is close to 1 year. Okay, so more directly, if we monitor the dystrophin protein, which live as in green fluorescence, you can see nicely. You can circular the muscle cell on the cell surface, and the disease model, and you can see completely gone of the green fluorescence. And the green fluorescence will be restored after treatment and lasting until almost 1 year, okay, 52 weeks. And at the same time, the syntrophin, this very protein, will work together with the dystrophin, which the gene actually is the wild type without any problem. But it cannot get onto the cell surface in the absence of dystrophin. But these guys will also be restored after the treatment. Okay, so you can see more clearly. I only show one kind of body weight to show the muscle strength. Okay, wild type versus pre-treatment. You can see the dramatic difference. Okay, because it's so weak of the muscle strength for the disease model. You can only the monkey, the etching model, can barely stand up, okay? But after treatment, the recovery is very evident, okay? So we have a different kind of body weight. I want to emphasize this disease model is very unfriendly, okay? And because it occurs in exon 5, and it occurred too early, okay? So majority part of this very protein is a foreign protein for this very disease model. Okay, so if you restore that, okay, and they will generate immunogenicity gradually. Okay, this is being confirmed by NEJM paper published last year. I won't get into any of the details. So in a parallel trial, we got 2 DMD primary cells from patients. Okay. And one is exon 49 plus 50, and another one is exon 48 and exon 51. So in either case, in both cases, okay, one thing we use iPSC to immortalize this very disease, the primary cells from patients, and then we start to treat using exon 51 skipping. Okay, in both cases, by calculation, we know if we skip exon 51, we have a chance to restore the reading frame. So in both cases, you can see the recovery of dystrophin protein. So this gives us a very strong confidence to pursue further. Okay, now we have a new disease model, which is the mutation occurring in exon 49 in NHP, and as I just said, after skip exon 51, we start to see dramatic increase, dramatic, downregulation of a CK and any other associated biomarker, which is way better than the exon 5 disease model. So for the sake of time, I'm going to quickly switch to another direction, which is base editing in mitochondria. So as we know, the mitochondria as an organelle, they have a lot of disease, mutation problems. On the bottom is kind of famous, all kinds of mitochondrial disease. And most of disease actually is because of a single point mutation. So, theoretically, we need, the base editing becomes the preferred kind of the tools for the correction, okay? We do have a base editing, which is the CRISPR system. But unfortunately, so far we couldn't apply the base editing based on the CRISPR system in mitochondria. The reason is because it's very easy to introduce the protein into the mitochondria, but not for the RNA, okay? There's a hindrance, the penetration is a problem. So that's why the whole field is being stuck for quite a long time. And obviously, you can think of alternative way using tail or zinc finger platform to generate base editing tool. But that's become very difficult. The reason is because the protein-based editing system, they cannot unwind double-strand DNA. So you just cannot deaminate, to do the deamination problems. Until 2020, I didn't include that slides. David Liu and his collaborator, they identified so-called double-strand DNA deaminase, okay, which is a DDA system. But the problem for that very system is they have interaction with CTCF to go back to the nucleus to generate, pretty broad spectrum unwanted deamination problem. So our strategy actually is to generate single-strand status by introducing nickase, okay? We figure if we nick one strand of DNA, we might end up with a transient, flipping of a single strand, not create a way to allow the single strand deaminase to work, okay? So we introduce a pair of tail, which one fused with deaminase, one fused with nickase, okay? Then we make it work, okay? So I'll skip all the optimization, some of the details. First of all, we generate this very model and system to achieve A to G kind of conversion. Then quickly, we can achieve C to T by swapping the deaminase, okay? And we can demonstrate we don't have this kind of DDA problem to associate with unwanted nuclear deamination, okay? So we generate disease model and so on and so forth. And we have just optimized our might be the system. And the reason we optimize is because one thing is we have two different kinds of direction for the MITO-BE. One is ABE, which is A to G, okay? Another is CBE, which is C to T kind of conversion, okay? So MITO-ABE is a problem, in terms of safety, is they have, unspecific RNA deamination problem. The reason is because we have to express the protein in the cytoplasm, okay? Then they have a chance to interact with RNA. Then they, cause this deamination in some RNA. So through the mutagenesis, my students identify one of the mutation, okay? And not only can get rid of all this unwanted RNA deamination, but also, increase, boosting the on-target efficiency for DNA deamination, okay? This is kind of a lucky break. For the MITO-CBE, the problem is different, okay? The problem for this kind of version is we arbitrarily use AppleBack1 as a deaminase in our first version. But this very deaminase have a TC motif preference, okay? So normally we want is follow the tail array to dictate where they locate and bind and to do the dirty work, not dirty work, to do the deamination work, okay? But they have TC motif preference, so this is the problem, okay? So the solution to solve this problem is very easy, okay? Because we have so many different kind of deaminase, which is CBE version. So we just pick one which works the best, okay? And many of them actually work much better than AppleBack1. But this is both safety and efficacy, is performed pretty well. Now we have a new version of the MITO-ABE and MITO-CBE. So I just want to use this slide to emphasize, because alongside this work, we generate circular RNA. We also make the vaccine. So we are kind of interested to compare mRNA as a delivery tool or circular RNA. And we identify in both cases, okay, in two separate independent cases, actually, circular RNA gave us much higher efficiency in terms of the mitochondrial-based editing, Okay, so this become a choice of our delivery. So for this model generation, you can see the comparison. Version 2 of MAT-AB have a higher efficiency in generate the disease model. And the MAT-CBE version 2, even more impressive because version 1, we just couldn't get it done, okay? But version 2, we could. Because we studied the generation from the germline of mice, you can see the tissue start from the very beginning. So we end up with the mouse disease model, which is tissue-wide, durable, and heritable. Okay, so indicating the efficiency of the mutation load is high enough to give us very stable kind of disease model. We can even identify the single mutation, okay, which is very important to have black-white kind of a correlation. So I just end up with this very slides to show we can generate 2 disease models: Lee syndrome, which have all kinds of symptoms, including heart rate, slower kind of symptoms, and LHON is vision loss. Okay, so I already used up my time. I plan to talk about another direction. I'll just in half a minute to finish through. And for the other direction for cell therapy, the essence for this very direction is a deep understanding of the mechanism. Okay, we have all kind of high-throughput functional genomics strategy to help us. So one direction is for universal CAR-T. We were able to identify a key gene, knockout of this very gene, which really can uncouple the HVG, GVHD, and also NK cell killing. And I'll skip all the details and just show you we have already done the IIT, show very promising and encouraging results. And a separate effort is on gamma delta T cell screening. We were able to identify a key player, which, the 2 kind of direction for the screening. One is the cytotoxicity, another is the persistency, okay? So we were able to identify 2 genes, okay? The combo mutation of 2 genes will convert gamma delta T cell to NK-like cell, indicating they will express a lot of NK type of receptor proteins. So that allow them to be more efficient and more powerful to go after AML cells. So this being demonstrated in both AML cell line and also the cells from the patients. Okay. Sorry for the time. I will close my talk by acknowledging my group, my collaborator, my funding, and if I have time for questions, I'll be happy to answer.
So thank you so much for Professor Wei's presentation. Any questions?
Thank you very much for the excellent talk. In Duchenne, have you considered to edit duplication rather than deletion? Because editing duplication could allow you to have a full and complete normal dystrophin, right? So it could be maybe also an interesting approach.
Yeah, there's all kinds of DMD, genotypes. Yeah, the duplication is just skip one and then can restore, right? This is certainly one of the target for this kind of therapy. Yes, I totally agree.
Okay, let's applaud again for his presentation. So the next speaker is Professor Wei Li. So Professor Li is director of Key Laboratory of Organ Reconstruction and Regeneration, Institute of Zoology, Chinese Academy of Science. And today his topic is technical development and the clinical translation of in vivo gene therapy. Let's welcome.
Okay, first I want to thank the organizing committee, like Sheila and Jiani, for inviting me here. It is my great pleasure to share some of my work. Okay. So before starting with my talk, I just want to briefly introduce my lab. My lab, as an investigator from Institute of Zoology, we face all kinds of animals. So we see the life as a persistent replicating system with extraordinary complexity. My lab study, one of the research focus is biotechnology and bioengineering. So we develop genetic engineering technologies and cell engineering, embryo engineering. And we want to converge all this type of technologies into a engineering biology platform, a mammalian-centered mammalian engineering biology platform. In one way, we want to understand whether and how we can regenerate the disordered part or injured part in our body. In the other way, we want to develop novel gene and cell therapies. I think for gene therapy, the concept has occurred almost 50 years ago. And this paper nicely illustrates the concept of gene therapy. So I think the gene therapy has been practiced a lot in Western countries. But in China, it lags far behind the developed countries. So in 2017, my institute asked me to establish a gene therapy platform. So I decided to discuss with doctors to choose a disease to start. Luckily, I met Professor Sui Fang from Xiehe Hospital and Dr. Wu Wenbin from Tongren Hospital and Professor Hu Danning. He's a founder of our country for genetic diagnostics for eye disease. So they suggested me to choose the Beatty crystalline dystrophy, BCD, because I asked them, we are a poor country, probably we should focus on a disease with a high incidence in Chinese population. So BCD, the occurrence rate in Chinese population, or in East Asian population, China, Japan, and Korea, 3 times or even higher than in West countries. So it is completely overlooked. Dr. Hu diagnosed this disease in China in 1980s, and almost 40 years, after 40 years, any treatment is available. So we spent some time to make the mouse models and knock out the gene. It's a single gene mutation, CYP4V2. We knock out this gene. The only difference is there's a huge difference, metabolic difference between the mouse and the humans. So we need to induce the phenotype with a high-fat diet. And if we feed the mouse with high-fat diet, the mouse will quickly exhibit the disease symptoms. So then we tested the gene therapy in this mouse model. Luckily, we found the gene therapy is effective for the mouse model. So we take another 3 years for translation. Now, it has been entering phase 3 in 6 medical centers in China. So today, the preliminary result shows the AV8 based gene replacement therapy is the mean average patient can the BCVA can increase like 3 lines, the 50 characters. They can see 50 characters more before the treatment. And the best dose, which we choose to enter phase 3, the intermediate does, they can increase with average 25 characters. That's 5 more lines for the BCVA correction. And also the functional assay to cross the barrier also shows very good result. Then we saw probably we can further, make a step further. Can we apply the gene therapy for not only for a single gene mutation caused genetic disease, but also for the complex disease like Parkinson's disease. So I think 6 cases of IT trial. The patient is very severe. This patient is 4 years old, but he cannot reach any movement milestones like raise his hand, which normally a child can reach it at 3 months after birth. So after surgery, 26 weeks after surgery, he can raise his hand, and then he began to learn to sit and to walk. So all the 6 patients after surgery showed a significant increase of dopamine in their brain and improvement of their movement. So we also transferred the same therapy into Parkinson. The efficacy study in monkeys showed that the therapy can also benefit the Parkinson's monkey models. So we transferred to, we applied for IND enabling. This is the phase 1 data. It's very preliminary. Now we only recruited 2 patients in Huashan Hospital in collaboration with Professor Wang Jian and Chen Liang. so 4 weeks post surgery, we can see the dopamine signals is immediately increased, and the 3 months later, the UPDRS score decreased 19, and the off wound also reduced, and the patient no any severe SAE events is observed. Since I think a big concern for the gene therapy is the accessibility. How to increase accessibility? One technical solution is to increase the programmability of the technology and to reduce the cost. So since most of the genetic disorders are loss of function mutations, so in most cases we need to introduce a therapeutic gene into the genome. So, but currently, although the rapid development with the genome engineering technology like CRISPR, but it only solves single gene mutations or small or large gene deletions. But for large DNA fragment, integration remains a big challenge in this field. Since it has such a big demand, so many labs are working on that. But currently, nearly all the published work relies on a DNA donor, because if you want to introduce an exogenous DNA fragment into the genome, you need a DNA donor. But DNA has many disadvantages. For example, it will induce very strong innate immune responses when it enters cells. For the RNA donor can avoid such disadvantages through chemical modification. That's the reason why it has been awarded the 2021 Nobel Prize for the COVID vaccine development. So we think probably we can develop a technology to use RNA donor to introduce DNA into the genome in a precision manner. So naturally, there is a natural system called R2 retro transposon. DNA transposon has been developed for more than 20 years. For the retro transposon, R2 is very unique because it can insert the target gene into the ribosome DNA loci rather than random integration. So the RNA loci can be considered as a safe harbor, so it's maybe suitable for gene integration. So we thought maybe we can develop R2 as an alternative strategy for gene insertion. R2 is, we use data mining to recollect 62 R2 system from the bird, the vertebrate genome, and we established a GFP intron reporter system in human cells to evaluate the activity of the R2 system. Only when the donor is reverse transcribed and reinserted into the genome, the GFP can be lighted. So using this reporter system, we test all the R2 retrotransposons. All the natural R2 transposons have very limited activity in human cells. The R2TG is a R2 system. The natural host is a kind of a bird. The R2TG shows a little activity. So we choose R2TG as a chassis for further engineering. Since it has two parts, one is the RNA as a donor, the other is the retrotransportase, a protein. So for RNA engineering, 4 parts are very important. The left homologous arm, the right homologous arm, and the 5 and 3 UTR. And we engineered these 4 parts. I will not go through the technical details. And for the protein engineering, we use different methods, including link single-strand DNA binding motif to the protein. And finally, we test the different combos for the final optimization of the R2 system. And after we choose the best combo, the finalized R2 system can reach 20 gene insertion efficiency in human cells. And now we increase the efficiency into 70%. And the system can be delivered in an all-RNA platform, an mRNA encoding the retrotranscriptase, and the other RNA code for the donor. So LNP delivered these two RNAs into the human cells, and then you can get a gene insertion in the human genome in the ribosome DNA loci. So the efficiency now in cell lines can reach very, very high, but in primary cells, in T cells, it only reaches 8%. In hepatocyte, it only reaches 20%. So it still needs further improvement to achieve clinical application. So we evaluate the integration specificity. That's very important because if it's not safe, it has a random integration, it will be not safe. Luckily, we use different methods to evaluate the integration. We found all the inserted genes are distributed in the ribosome DNA loci, but not in other genome loci. And the integration has little impact on the cell behavior, cell survival, and transcriptome. In each cell, around 1.5 copy is inserted. So there are around 200 copies, ribosome DNA copies in one cell. So 1.5 is pretty less. So it probably will not cause severe interfere to the cell state. So now we are still improving this system. If it works, we hope we can develop an RNP mRNA-based gene therapy for all the genetic disorders. Okay, so I will stop here. Finally, I want to thank all my collaborators and students, trainees in my lab, and thank my clinical collaborators and all the patients. Okay, thank you.
Okay, thank you so much, Professor Li. So any questions? Okay, in the interest of time, let's move on to the next session. So there will be a panel discussion. Let's warmly welcome all the speakers again onto the stage, and also Sheila from Cell Stem Cell to moderate the panel discussion.
I see.
Okay, well, thank you, everyone. We've reached the end of our day, and we thought we'd have a conversation to kind of integrate and wrap everything up. So I think, well, the first question kind of takes us back to the morning when we were talking about cell therapy. And so the question is, how can the lessons that we've learned from CAR T-cell therapy in autoimmune diseases and rheumatologic diseases, how can that be applied to advance cell therapy in other areas of medicine, such as neurodegenerative diseases and metabolic diseases? And so I'll turn the first question to Huji.
Thank you. That's a very good question. And the simple and straightforward answer, I believe, is yes. The lessons we learned from biologics we use quite a lot over the last 20 years for treating the autoimmune disease. And we show the biologics can be also used for the other diseases. And actually, efficacy is pretty good. However, when we see the CAR T cells and we published in Cell, also the George's paper, we can show that the efficacy CAR T cells actually is better than biologics in the autoimmune diseases. And so similar approach, I think they can also use for other diseases. But the big challenge and the question we face now is, where's the target? So that's the question. But I believe sooner or later we will find the target. Once we have the target, I'm sure they will clean the target cell much better compared with the biologics. This is already shown, and in our data, also in the George's data, you can see that they can clean the target cell more deeply, and even in the tissues for the target cells, like in lymph nodes and also in the skin, which was never seen for the biologics before. So probably that's the reason the efficacy is better. So as I said, target, what's the target? And whether or not it can be used for off-label, or we really need to find the right target for other diseases. And we believe that the target must be, play important role in the pathogenesis of the disease. Otherwise, not going to work. That's my answer. Probably you can add more.
I think I absolutely agree with what you said, and many, many points and many aspects of that I would exactly put that way. I think what we have not fully understood is also that we have additional cells that we can engage. So we have not been talking much about Tregs. We talked about over lunch here in the panel. We can think about macrophages and really different ways of other cells that are used to target, but also to deliver. So we could think about metabolic reprogramming induced by cells that we put in the system like Tregs, right, that not only can modulate the immune system, but also could modulate potentially the metabolic situation at a site where we hopefully have to identify targets first. So I think the possibilities are nearly endless. And I think we just have to understand biology and also the targets better, and then we can attack much more precisely.
Dr. Kim, maybe we can hear from your perspective the other angle.
Yeah, Sheila, thank you for asking that question. Really, I appreciate it, because ever since we published our needle trauma paper last year, your question is my question, actually. I've been always thinking about it, because I didn't have time to share all the detailed data, but we found the needle trauma is a very serious issue for all kinds of brain cell transplantation disease. And we found that needle trauma could be a very fundamental issue. And then we found that the T regulatory cell can diminish the needle trauma and increase the dopamine cell, but I didn't have a detailed explanation. What we found is that dopamine cells after transplantation, more than 90 to 95% die. When we co-transplant the Treg cell, it can increase the improvable only 2 times. In other words, only 20% at most can survive, but still 80% die. That's why I'm saying that my question is, how can we enhance the power of the Treg cell so that they can completely control both adaptive and innate immunity? And after that, all the papers I'm reading now is the immunology papers, and I really learned a lot from these two expertise. And in the coffee break, I learned a lot again from Fabian. And the more and more I realize that we have to interact, the neuroscience and cell therapy people also interact with the immunologists and exchange our wisdom and problems so that we can help patients much better. So I absolutely agree your point, and we really need to further discuss about and communication. Yeah.
Yes, thank you. Immunology is everywhere.
Absolutely.
Okay, and we'll turn the next question.
Okay, next. So I have one question, mostly for Professor Li Jianhui and Professor Jiena. So the question is, what are the key challenges in translating bench research on cell converting and tissue regeneration into effective clinical therapies at the bedside, and how can the challenges be addressed? So.
Yeah, so I think there are a lot of challenges. So end of my talk, I list a lot of technical challenges. So and I think MOA is for sure very important if you want to really improve the outcome of your therapy. The MOA is one of the first thing you have to think about as a biologist. And another thing I will say very important nowadays I realize is CMC. So when we work in the lab, we control everything what we can control in the lab. But if you want to translate it as being a medical product or as a drug, cell as a drug, gene as a drug, you have to really think from the angle of pharmacology side. So it's a very very different idea of how you develop the whole what you find in the lab. So I realized that not every original findings can eventually be end up with as a effective medicine. That's the reality we have to face. So at the beginning, I'm saying like very beginning, you have to think about whether you can bring that to be the medicine. Because the medicine, you have to think a lot about the regulation, about the quality control, about the safety issues, and all these things can affect, I'll say, like whether the original findings can be the effective medicine. And I think that's a very long way compared to the original findings in the lab. And that's, I think, probably the second most important challenge. And final thing I will say is the funding. So if you face the industry, then you will realize the money required for developers as such thing is, I'll say, 10 or 100 or even 1,000 fold compared to what you consume in the lab. So these are the challenges that people have to face. Of course, in the future, when I go through with all these things, I will realize other challenges. This is so far my experience, so I can share with you guys. Yeah.
Thank you so much.
Okay, I very much agree with Professor Hui. And in our research, I think, for example, our research about using endothelial cells to repair eye choroid ischemia, I learned a lot. First is an area that nobody have paid very much attention to before, but then my clinical colleagues come to me with this situation, and I think it's as a basic researcher, we should keep an open mind and looking for the best scenario to apply our technologies and talk more with our clinical colleagues. So I think that might be maybe an interesting, unexpected way to use our endothelial cells. Maybe will be the first therapy can move to clinic, and then now we are trying to translate this Also, we run into a lot of practical problems, like funding, like also because this is a very, very new therapy, so there's no examples. So we basically have to set up standards and find new ways of delivery, and everything will be new. Yeah, but it's an interesting course. I think there'll be more hope in the future to address many unmet clinical needs with cell therapy.
Okay, thank you so much for the insightful thoughts.
Yeah, so I noticed that many of the speakers have founded companies and consulting many. So in that regard, I want to share a little bit about my experience. My research is cell therapy, but also another line of my research is drug development for the Parkinson's disease. I don't have time to share it, but we identified the drug target, and after long years, we finally generated a drug candidate, and then now I also founded a company. But my experience is running the company is that, of course, doing good experiments and publication and get funding from NIH is very challenging. But when I started company, I realized that the real challenge is get funding for the company. So in my case, when I tried funding from Korean investors, they say that, why don't you get the investment funding in USA? But then in USA, it was also not easy because relatively I don't have network because I finished all my education in Korea and then I came to United States as a postdoc. So relatively I don't have a big network. So it's very difficult. But luckily we just finished the phase 1 clinical trial and we are moving to phase 2. But I think our speakers don't have time to share their experience about founding the company and running the company. But in my case, that was really, really challenging. So when young people, successfully do the science and get innovation and they want to commercialize it, how to commercialize it, that's a big, big challenge and obstacle in my mind.
Oh, yes, thank you for sharing that perspective. And I do think it's important to think beyond the science and what is going to be needed in order to bring the therapies to our patients. And so the next question kind of turns again away from the scientific questions and more to ethics and regulation. So first we thought we'd ask the European perspective from Dr. Servais and then the Chinese perspective from Drs. Kai, Wei, and Dr. Li. So our question is, for advancements in gene and editing technologies and cell therapy, in order for those to get translated to clinical applications, what do you think are the most important ethical and regulatory considerations that need to be addressed, including how to overcome the cost issues? So first, I'd like to hear from Dr. Servais, extending from your interesting talk today, and then I'd love to hear the Chinese perspective and see how that might be different.
I think that for me, the very first aspect, when it comes to apply a technology, which is at the beginning disease agnostic to a set of disease, is definitely the ratio between the benefit and the risk, right? I mean, personally, I felt quite comfortable to conduct trial in SMA type 1 because these patients were dying in a couple of months. We just need also to appreciate what is the risk when you consider such population. And the risk when you consider patients who could die in a couple of months is to transform these patients into patients who live for a couple of decades, but with a very poor quality of life. So I think that it's extremely important to engage first with, of course, physicians, but also with patients advocacy, with patients to understand what is the specific risk, what are the fear in the specific population that we are trying to address. So we see that it's certainly the understanding of what is the risk. Dying when you have SMA type 1 is not a big risk. The big risk is surviving in very poor conditions. And that's very different, of course, when you consider a disease eye, right? Where you lose vision, dying become a much higher risk when you are 30 years old. And then that's the first thing. The second thing is, in my opinion, transparency. Transparency and honesty in the way we address and we share the informations from science and from clinical trial. I think that one of the big, and this maybe is a bridge with what we've discussed about the companies, but one of the big issue I think today is that companies, as they are on the stock market, have to communicate simultaneously with everyone, which means that the communication is not peer-reviewed, and the communication is even not during a congress. It's a press release. It's a press release and results that are presented by the CEO or the CMO of the company, and we invite a lovely investigator to say, well, I've included patients and it was okay. But at the end of the day, the data are not peer-reviewed. We don't know exactly if we can trust it or not, but they are on the on the internet, and you can read it from anywhere in the world. And this raises what I've discussed in my talk as sometimes the unrealistic expectations. So that's for me the two main issues from an ethical perspective, is to appreciate what we call risk, and this is population-based and needs to be consulted with the patients and the patient's advocacy and the physicians in charge, and be the transparency and honesty and clarity when we communicate our data.
Okay, I think for me, I think especially in developing countries, safety issue is always ranks first, because people lack basic knowledge of advanced therapy, and if so many ethical problems are induced by safety issues first. Of course, there are many other ethical considerations, but firstly, the safety issues is very important, especially for advanced therapy like gene editing therapy, because compared to other treatment, it is more irreversible. So if something bad happens, it is very hard to stop the treatment. So we should be more cautious about such types of therapy. Of course, how to balance the benefit and risk is always under debate, but we should, all the people involved should put the safety issue rank the safety issue as first. For ethical, other ethical considerations, I think for example, in China, how to authorize for those people with highly educated or with receive good education, it's good to discuss based on some public knowledge. But for some people from poor countries, sometimes they try to sometimes how to achieve the transparency. Sometimes it's difficult. So in such situation, probably we need more organizations, like third-party organizations, not the company, not the scientist or doctor, like other organizations, to broadcast the basic knowledge to them. And I think that's the best way in developing countries to achieve transparency, because there's no authorized organization to say it's good or it's bad for advanced therapy, for improved treatment. Yeah, okay, that's my thought.
I think overall in China, the policy encourage to do new clinical trials, still there's something you can do. It's like a red line, for example, a germline modification. That is something you can't do. But the rest thing you can do. But still there's something you are encouraged to do. For example, there's some disease that there's no existing drugs. For those diseases, I think it's a good choice to start with, because there's no drugs available. The patients need new treatment urgently. And for the CDE, which is the FDA version in China, they are getting more and more open for new clinical trials. In past, the CDE often approved new IND application after FDA, but now they are more open. They are starting to accept new IND before FDA. So I think the overall environment for new drugs, new therapies are getting better and better in China.
All right, if I can add more, both of them cover most part of the ethical issue in China. But for me, actually, one kind of red line ethical issue is like germline editing, which is kind of straightforward. We have this very strict rule not to get into this kind of direction. But for therapeutic ethical issue, for me, actually, it's a really balance between benefit and the risk. And, of course, both is important. But if stress more on the safety, then it's very hard to get anything approved, okay, in time. But if you emphasize on, you want to have a chance, especially for most of the scientists who invented technology or, have the new way to do things, of course, they want to or eager to have this, practice or get into the patient. But the safety, oftentimes it's being overlooked. They might generate problems. So I think it's a fine balance between these two, and it's very important for ethical issue. I think it's important for like hospital or clinical center who take the responsibility to really safeguard and to prove in a very scientific and timely manner. But really, it's really case by case. And I agree with most of you guys, the so-called transparent. But for me, I think it's kind of difficult to really practice so-called transparent, okay? If you open to general public to get into the discussion, oftentimes this will lead to, chaotic kind of situation, right? So I don't know what's the best solution, but I only want to say it is important to balance these two. So, leaving the ethical discussion for the cost, I think for me, it's important to have the technology development, especially locally. If you're talking about the China perspective, I think it's important we have some foundation technology invented, or even the following up technology, which give us a way in terms of price. We are sensitive in the price because we are not developed country, first of all. But we don't want to, for others, for many of you guys to have the kind of impression, whenever, Chinese scientists or industry get involved, we will, basically, break down the price to, as cheap as possible. Which is not necessarily the case, and we have the need to lower the price. I mean, not only in China, okay, in the United States, in UK, and in most countries, obviously we want to have affordable new therapeutics, option. This is applied to every country, but I think it's a collective, effort to really bring down in a reasonable manner. This probably have to get involved the government effort, the CDE, FDA, including the insurance policy. So I think it's a collective effort to really make it reasonable and affordable. So this is just some input.
No, thank you. I'm glad you spoke to cost because perhaps this is a naive question that I have. I'm sure it is naive. We heard earlier today how cost can drive inequities, right, in access to therapies in Europe. I'm curious, how does cost affect the access to therapies among Chinese patients and the insurance situation here?
Well, if you ask me, I think it's kind of a tough question to answer. Some of the therapies, like CAR T cells, have already, some get approved in China, and still it's a pretty pricey kind of therapeutic options. And that's also why many of the allogeneic strategies is really being chased, and it's a very important, especially in China. But for many other new technology, mediate kind of new therapies, I think it's still very early to say anything because almost none of them, other than CAR T cells, get really into the market. I think the insurance or policies obviously so far is lagging behind, but most of the agency and including the governors, I think they are aware of that. So hopefully they are in the process to get things done in time to meet the new future challenge. This is only my biased opinion. Yeah.
Thank you. I think we'll turn to our final question.
So I'd like to ask a final question. So it could be quite broad, but it's open for all the speakers. So considering the global landscape of cell and gene therapy research and development, how can international collaboration and knowledge sharing be enhanced to foster innovation, address common challenges, and ensure equitable access to the latest advanced therapies across different regions? So it could be from any perspectives, not limited to Chinese or Europeans, so any thoughts are welcome. Yeah.
Okay, well, I will start first. Okay, I think to share knowledge is the most important. I just take example, when I published my paper, I just introduced the R2 retransposon paper. I immediately received two discussion invitations. One's from Denmark, the Novo Nordisk company. The other is from American startup, Tessera. So I think currently sharing knowledge as a scientist is very big contribution to the collective efforts to advance the cell and gene therapy. Okay.
I actually wanted to say something very similar, but points that you just raised are also very important. So events like the one that we just have here today is really the critical thing that has to happen. I think sharing knowledge, not just with companies, but also among academia, like our peer, is really crucial because we are usually driving knowledge into like the common sense, right? And academia is different in that aspect from companies that typically want to keep their new information to themselves because for them it's IP and intellectual property that they want to develop further. So I think exactly like we do here, sharing our experience, sharing our thoughts and the questions that we have to then address them together is really what drives science forward. So I can only encourage you to keep that meeting up. It was really lovely and a big exciting event for me. So thanks again for having us.
So in my experience, in the Parkinson's cell therapy field, we had the very interesting symposium, so-called Global Parkinson's Disease. So we had the active group working on the Parkinson's disease cell therapy. We had an annual meeting. So it was very insightful and helpful to share what's going on. So that was one example. Another example I want to share is that in terms of the collaboration, I think one of the most powerful ways is to exchange scientists. So I have several labs where we exchange our researchers. So usually graduate students come to my lab and stay 1 or 2 years during their thesis work. So that can be part of their thesis work. So that can be most effective way of collaborating and sharing. Then lastly, collaboration and sharing information is a little bit delicate issue. It's just like the clinical benefit versus risk, something like that. But if we are very wise and strategically form the collaboration, that can be very synergistic and powerful. So, depending on how we manage it and what kind of a system we have, this is really powerful way to advance science and clinical application. So I like to suggest that we, scientists and the editors and also governmental officers work together to make a very strategic and wise program to enhance this kind of collaboration.
So I think, so I grew up see the relationship between China, America, and China and Europe, very good relationship growing. But after the COVID, since the COVID, I think the intention, the competition is very clear to me. Like at least I feel. I think I need to say this concern. Like I was in America States 2 weeks ago. I talked to many scientists, concerns from different people. I think that's the biggest barrier nowadays. So I don't know. Like this is the same thing we can at another level of barriers like. So for us, I think at least I think exchange ideas, collaboration is very important. And the other things I think countryside, something I hope the best. That's all I can say. Like, but we will still keep going on. Yeah.
Thank you. Well, I would take my cap of rare disease guy. I think that we need certainly to move forward to be able to identify who are the patients who are probably the best responders. Because all these therapies are obviously amazing, but I'm not aware of a single therapy after having injected 100, 200 individuals for which you don't have good responders and bad responders. So it comes then to two aspects. The first one is the proper identification, and when it comes to rare disease, I think that the early identification of the patient is key. And I will then bring a concept that we've not discussed yet, which is newborn screening, and which is the identification of the right candidate, those who are really at the very early stage or even before the onset of the symptoms. And I think that it's this bringing the therapies as early as possible in the development is key, because we all know that in neurodegenerative conditions, it is going to be much more difficult. So identifying the right group of patients, and earlier is better. So early diagnostic, screening, and pre-symptomatic is sometimes the key. That's what we have observed in SMA. And then I will take the opportunity that we have the editors in the room. I think that to transparently report negative results is very important. I think we do not see enough negative trials and issues in the papers. And that's a problem. Because today, for instance, and I see it in my field, for instance, in Duchenne, idebenone was developed in Duchenne, and we have a successful phase 2 trial that was published in the Lancet, and the negative phase 3 trial was never published. Which means that we all know the story, but we're getting older and older. So the people will read the paper soon will forget that there was a phase 3 trial that was negative, and it's impossible to find. And this is one example, but there are many others. And I think that it's very important to publish negative results, to give them the same weight than positive results, to publish outliers, and to to respect the outliers, not trying to cherry pick to increase the impact of the journal. And this is a collective responsibility. We as author, or also as reviewer, rather than pointing out the outlier, to say, well, the authors acknowledge there is an outlier. They try to explain. Maybe they don't have the full explanation, but at least they mention there are outliers, and they have investigated as much as they could. But again, I think that it's all a matter of collective responsibility to properly, report our data, including the negative data, including the outliers, including things that did not go as we were hoping they would.
So everyone have to say something. That's all right. That's okay. I said, and I believe that sharing the knowledge and the collaboration is essential for science. That's no doubt. We benefit from these lots. We have large consortiums for ankylosing spondylitis and across the world, from European, China, and Korea, and North America, and we actually make progress very fast for this disease, because genetically this is very complex, and also different ethnic background have different way to handle this disease and dealing with this disease. So the sharing the knowledge and also collaboration really benefit to all the community or the scientific community. So, but I believe that we all know that they have some issues now. So, in this way, I believe that the scientific community, including scientists and the press, journals, we should working together, regardless, politicians what they think. So we should create the nice environment and help the science eventually help the world. That's what I said.
As far as I can remember, before COVID-19, US, EU, China are all very happy. Then suddenly the COVID-19 comes, the collaboration stops. I think the only solution to go back to those happy days is via collaboration. And I think collaboration will bring mutual benefits, and it will make cell and gene therapy drugs quicker, cheaper, and better, and the benefits economies in all countries and benefits all those patients.
I'll use both. I cannot offer any different answer for these questions. Obviously, probably we all agree, communication, collaboration is very important. I think if any of us, sitting in the audience, you want to learn from your colleagues, probably you realize you want to be a good community members. You want to offer your insight, your perspective, your results. I think it's very important. The bottom line actually is we have the common enemy, which is the disease. We want to, it's not this disease is unique for China. Of course, we have like a regional kind of a focus, but we have common disease problem to solve. So to unite together to really, it's the best way actually to share the information is very important for academic scientists, and to collaborate, to join force is essential to really to move things forward in a most effective way. So I can only wish the politics, the conflict between countries will never get in the way in the academic, collaboration and the communications. So, yeah, thank you.
I think international collaboration and exchange and discussion are very, very important especially for cell therapy, especially for stem cell derived cell therapy, because we need to have a consensus or a standard for the cell function, the cell type, the cell characteristics, and this can only, I think, be reached, be accepted with like a more international collaboration and a dialogue and continuous optimization.
Before we finish, I'd like to again thank Sheila and the organizing committee for organizing this very special meeting. Actually, I felt that this meeting is very special, and I learned a lot. But I have to confess that 3 weeks ago, I had the time to visit Korea and give a talk at the cusp and Seoul National. And I asked my colleagues, nobody knows about this meeting. So I think the only thing is maybe you better advertise this meeting a little bit so that the more people can come. Definitely, they should be aware of this. And because this is a very exceptional and very stimulating meeting.
Well, thank you. Thank you so much for those comments about this meeting. And this will be available online, and I believe we'll have over 1 million views after some time. And I really do appreciate your comments on how journals can help facilitate collaboration more, really take that to heart. So and also just the spirit of this meeting of bringing people together and transcending any political differences. As you said, we have a common enemy, and it's the disease. So I think we'll now close our panel discussion, and I'll turn the floor over to my colleague, Duk Lee, to say the final closing remarks for our meeting. Thank you so much.
All right. I'm very privileged to have these last words, and I'll try not to ramble too much, because I'm aware that I'm the only thing that's standing between you and the relaxing evening and the dinner and the weekend. So I'll try to be thoughtful. So first of all, we have a long list of thank you, which kind of nicely dovetails into the last speakers. So I would like to extend our thanks to the generous sponsorship from BMSTC, and special thanks to our host, Professor Qi, the Dean of Tsinghua Medicine, Professor Wong, and other colleagues at Tsinghua University for hosting us in this wonderful, historical, beautiful building. And thanks to all our colleagues at CellPress, especially Ludi, Brenda, Joy, and other marketing colleagues in China for all the logistic coordinations. And I'm aware that there's millions of tasks to be handled at the same time, and plus the technology as well. So thank you very much. And thanks to Sheila and Jiani for co-moderating the whole event and also the panel discussion. And of course, a big thank you to all our esteemed speakers from China as well as from abroad, and especially for those who have to fly a very long distance to attend this event, and also for those trying to participate online despite the time difference as well. And thanks for all of you for sharing your thoughts and your works. And finally, great thanks to our audience for actively participating in the discussion and in the questions. So that's my thank you. And so I'd like to reflect a little bit on the content of the event. It's a remarkable event. And so when I think about patients with a tumor, for example, our first initial approach would be to cut out the tumor. And if it doesn't work, then we throw in a bunch of toxic chemicals and radiations. And so that surgery and chemotherapy and radiotherapy, that's the 3 cornerstones of cancer treatments that's been there for centuries, back in the 19th centuries. And then the 20th centuries, we saw the emergence of targeted therapies like TKI, small molecules, and then the groundbreaking work of the immune checkpoint inhibitions discovery by the 2 Nobel Prize winner, and then I still remember in early days of my editor careers back in the 2010s when I attend conferences, there's big buzz about the anti-CTLA-4 ipilimumab and the anti-PD-L1 nivolumab. And then within just over a decade, now we have over dozens of immune checkpoint inhibitors. And they themselves established themselves as the new cornerstone for cancer treatments through pivotal trials like Keynote and Checkmate. So today I'm very super excited to see the remarkable progress that each of you have made, contributed to the field of cell and gene therapies, from the groundbreaking research on the new technologies through the final products that are ready for clinical use. And of course, I'm just not only just using cancer as an example. Your discovery, the implication of your work is beyond that. And today we've seen the progress made on CAR-T therapy for autoimmune diseases, cell and gene editing technologies for tissue regenerations, for neurometabolic disorders, Huntington disease, muscular dystrophy, beta thalassemia, just to name a few. Just now, our panel have discussed the challenges and the opportunities that we're facing in this field of research. And as we are moving this field forward, I'm constantly, acutely reminded about these three recurrent themes that we've heard throughout the day. So the first one is do no harm. So we should all bear in mind that Hippocrates' oath, whatever we do to our patient must not fare worse than they already do. So patient safety comes first. And then second is efficacy. So the bar is set so high, higher than ever. Then I come back to the cancer example with the immune checkpoint inhibitors, and there are various combinations with other modalities, so especially in the neoadjuvant settings. So our innovation in cell and gene therapies have to be even more efficacious than the existing standard of treatments. But that's also very exciting, isn't it? So then thirdly comes the cost. So innovation is expensive. And the success rate is very low. We all have our own driving motivations. As a scientist, we innovate because we are curious. We innovate. We want to know new ideas. We want to test new methods. Blue sky research is expensive, but it's important for human knowledge. But remember those Nobel Prize laureates. The foundation research serves a purpose. It needs to improve human life. And in the disease scope, it needs to reach the patients. So if a fancy treatment is very costly, then its impact on patient is minimal. But I'm very hopeful because I see that we're all open to collaboration, open to each other, to share our findings, to share new tools, new knowledge. We're open to collaborations, to a healthy competition. So I train martial arts, and my Sifu always told me that I need to be better than my yesterday self, and don't worry about beating my opponents, because you cannot beat everyone. So I believe that if we focus on our own missions, to improve our own tools, to treat the patient in front of us, rather than worrying about the guy in the lab next door, then we will get there faster. So I'm hopeful because we're moving very fast, faster than ever. So when we see that regulatory has to catch up with technologies, I think that's a good sign. So I'm hopeful, actually I'm quite confident that in my lifetime I will see cells and gene therapy to become the new cornerstone of disease treatment. And that's thanks to all of you for your scientific discovery, for your commitment to advancing healthcare, and for your collaborative spirit. And I hope that this event has ignited motivations and fulfilled your inspirations. And enjoy the rest of the day, and have a wonderful weekend.